It's finished.
And you're saying to date there is no pictures of purified virus?
Today, there is no pictures of purified virus. And certainly, Montagnier did not publish it, Gallo did not publish it, Levy did not publish such pictures, Weiss did not publish such pictures. And the only pictures which have been published was, in fact, this is admitted by the Franco-German researchers in 1997, when the first attempts, the first pictures of what is called purified HIV were published by two groups, one from the United States and one in a Franco-German study.
You said that in 1997 they did try to purify HIV, is that correct?
Yes.
And you're saying they weren't successful?
They are not successful. And what is more important, these authors, they accepted or they admitted, but by 1997 there is no evidence for purification. And here it is. This is from the Franco-German study. Virus to be used for biochemical and serological analysis or as an immunogen, that is as an antigen, is frequently prepared by centrifugation through sucrose gradients. And they said, in none of these studies has the purity of the virus preparation been verified. So by 1997, there is no proof that HIV has been purified.
But did they purify HIV?
They tried. So they accepted this. As I said, we've been asking this for the very beginning to have some evidence for HIV purification. And these authors tried to present, or they did their best to purify HIV. And here is their evidence. This is the Franco-German study. And they had material which is meant to represent purified HIV. The top and the middle is material obtained from infected cultures, and is meant to represent purified HIV. The bottom part is material obtained in a similar manner from non-infected cultures. Mm-hmm. And as you can see, the arrows, they said, represent the HIV particles. First of all, this cannot be said to be purified virus. As you can see, they are not purified particles. In fact, only the particles which they put the arrows are said to be particles which look like HIV. The rest are all cellular fragments, or called vesicles. In the material which is obtained from the non-infected cultures, you can see even there are some particles which may look like the one which is arrowed. So it is significant that the authors do not call this material purified HIV. In fact, they call it purified vesicles from infected H9 cells, the top, and activated cells. So the authors admit that this cannot be considered purified HIV. With all the effort they put, they could not obtain purified HIV. Okay. It is also important that the particles which are arrowed as representing HIV, they don't have all the morphological characteristics attributed to HIV. There is no evidence for knobs. In fact, even their diameter is higher than what is considered to be the retroviral particles.
But does size really matter? I mean, humans vary in sizes.
They matter because this in the Franco-German is the size is they are larger, but not as large as in the American evidence for purification. Here is the effort by the American researchers in purifying HIV. These are their results. And as you can see, they were not able to obtain purified HIV. And again, the particles which are arrowed and are said to represent HIV, they do not have all the morphological characteristics attributed to lentiviruses. And in fact, they don't have the knobs, they don't have cone-shaped core, you cannot see there, they don't have lateral bodies, which are, they should be in HIV, and most importantly, their diameter is they're too large. The average diameter of the particle is 234, and none has a diameter less than 160. Just by taking their diameter, it is impossible the particles which are labeled as HIV to be HIV.
Is the size also important in determining what you're looking at?
Yeah, think about viruses can be as big as pox viruses, up to 300 nanometer. HIV up to 150. The smallest autonomously growing virus will be a circovirus, just 15 nanometer.
So what's the smallest HIV particle that's ever been documented?
I think it's something 120 to 150. No, these are fixed morphological entities.
They don't change.
They don't change.
So that's a huge thing in helping you differentiate whether it's HIV or not.
Yeah. The size of a structure is very important to make the diagnosis.
Now, if the particles which are labeled by Bess and his colleagues as being HIV, then the absolutely necessary condition is for the material which was obtained from the infected cultures to have proteins which are not present in the material which was obtained from non-infected cultures.
Right.
But this does not seem to be the case, and in fact, Bess and his colleagues have come with this evidence. They took the proteins from all the three bands they had, from the two infected cultures, or what they called infected cultures, and from the non-infected cultures. And here are the proteins from the non-infected cultures, which they are put in a strip. Here are the proteins from the two infected cultures. As you can see, if you look at this, there is a difference, but the difference between the strips is only quantitative. That is, we have these bands in all the strips, only there is less here. Similarly with all the other proteins. In some of them, in fact, they are exactly the same. So by looking at these pictures, the proteins which existed in the purified material, the HIV purified material, and in the material which was obtained from the non-infected cultures contain the same proteins, which means that none of these cultures contained HIV. As I said, if the cultures which contain HIV, they must have had proteins which are not present in the non-infected cultures. And yet, this is not the case. The proteins, all the proteins are found, which are found in the so-called infected cultures, they are also found in the non-infected cultures. Some of them in smaller quantities, but nonetheless, they are there. The difference may be just because the way the cultures were conducted. The American authors labeled some of the proteins, and the P6, P7, P17, and P24 are labeled as HIV proteins. And these two proteins, that is, proteins which are around 32 and around 41, they were labeled as cellular proteins. No label is put in the proteins which had molecular weight higher than 41. Yet there are many HIV proteins which have molecular weight. Now, why they didn't label them? First, all the proteins which are around 41 and 32 are non-HIV. There cannot be HIV proteins with molecular weight 41 or around 41, a molecular weight of 32 or around 32. Okay. We asked Bess why they labeled these other three proteins as HIV. And he said that they put this label because that's what the reviewer asked them. But they did not have evidence themselves that they were HIV proteins.
He stated that they did not have evidence that they were HIV proteins?
Yes, he stated that they did not obtain themselves, but the reviewer asked them to label them as HIV proteins. And he said the reviewer was right. We labeled them HIV.
Did he specifically state they had no evidence those were HIV proteins?
Personally, they did not have evidence. Yes.
Bess said that?
Yes. In this experiment,
Debemos recordar que el test jamás detecta el propio virus, sino los anticuerpos que han reaccionado a la presencia del virus en el organismo. ¿En qué consiste el test del SIDA? Se desarrolla en dos etapas. La primera es el test ELISA. Si el resultado es positivo, debe ser obligatoriamente confirmado por una segunda prueba, el test de Western blot. El Western blot consiste en una bandeja sobre la que han sido fijadas las diferentes proteínas del virus. Dicha bandeja se pone en contacto con el suero del paciente. Si el suero contiene anticuerpos contra el virus, las proteínas aparecen en forma de bandas negras. El test es positivo cuando la bandeja presenta varias bandas negras. Hasta aquí todos de acuerdo, pero posteriormente las cosas se complican porque los criterios valorativos cambian de un país a otro. Francia no tiene los mismos criterios que los Estados Unidos para aceptar una prueba positiva. Dichos baremos varían también en Australia o en África.
En estas columnas podemos observar los distintos criterios que se emplean para definir un test Western blot positivo, es decir, para establecer cuándo una persona está o no infectada por el VIH. Los criterios que se aplican varían en cada país e incluso en las distintas instituciones de un mismo país. Si aparecen dos bandas negras en el test de un paciente, sería considerado seropositivo en África y en algunos estados de Norteamérica, pero no lo sería en Australia, ni en otros estados americanos, ni en Francia. Si aparece una tercera banda, una P24, no será considerado seropositivo en Australia, ni en muchas partes de América, ni en Francia. Si aparecen cuatro bandas, tampoco se les considera infectados por el VIH ni en Australia ni en Francia.
No puede considerarse infectada a una persona aquí y allí no. O se está infectado por el virus o no se está. Con estos criterios no vamos a ningún lado.
No es fácil encontrar una explicación lógica a la arbitrariedad de estos baremos. Se deberían aplicar los mismos en todas partes. ¿No sería más sencillo y más seguro? Pero los laboratorios no son de la misma opinión. La sangre contaminada, dicen, sean cuales sean los criterios aplicados, reacciona positivamente y siempre aparecen todas las proteínas del virus. En ello radica la diferencia con la sangre no contaminada.
Aunque se unifiquen los criterios, seguiremos teniendo el problema de la imposibilidad de repetir un mismo test. Es muy difícil hacer dos tests idénticos. Por ejemplo, este test de una misma sangre ha sido realizado por 19 laboratorios norteamericanos diferentes. Los mejores laboratorios, los más prestigiosos.
Cada bandeja corresponde a un laboratorio distinto. En algunas aparecen varias bandas negras, en otras tan solo algunas. En la bandeja del centro, en la letra I, no aparece ninguna. Pero el suero pertenece a un mismo paciente infectado por el virus del SIDA. Los laboratorios australianos han detectado errores aún más graves.
Para intentar explicar el porqué de estas enormes diferencias, me gustaría mostrarles el estudio realizado por los analistas norteamericanos. En 1991, El Diario Americano de Patología Clínica envió 101 muestras negativas, es decir, que no tenían bandas, de sueros Western blot, a 3 laboratorios distintos. En 67 muestras contestaron que no aparecían bandas. En 13, que aparecían bandas de distinto tipo. En una, P41 y en otra, P24. En 2 muestras, P24, 32 y 41-120, lo que para muchos laboratorios se considera un resultado positivo. Sin embargo, todos los sueros eran negativos.
Es decir, que el análisis de un suero puede resultar positivo en un laboratorio y negativo en otro. Los australianos opinan que los resultados contradictorios ponen en entredicho la validez del propio test, su capacidad para determinar si una persona está o no infectada por el virus, lo que los científicos denominan especificidad.
La especificidad de un test asegura que el resultado sea positivo solo cuando el virus está presente en la sangre. Si el virus no está en la sangre y a pesar de ello hay un resultado positivo, el test no es específico.
Los australianos han llegado a afirmar que los resultados positivos en las pruebas del SIDA no confirman de manera absoluta que una persona esté infectada por el VIH. Para comprobarlo, han realizado pruebas de SIDA a ciertos animales que no sufren la infección por el virus. Por ejemplo, a estos ratones, a los que se les ha inyectado sangre de otro ratón y se les han encontrado anticuerpos. También a estos perros. De los 144 perros utilizados, la mitad presentó los mismos resultados que los ratones. Se han realizado pruebas a partir de sueros extraídos en 1968 a indios amazónicos. Muchas de ellas dieron resultados positivos, a pesar de que en esas tribus no se ha producido nunca ni un solo caso de SIDA. Lo mismo ocurre entre algunas poblaciones de ciertas regiones de África. Los virólogos que trabajan con el virus del SIDA están acostumbrados a este tipo de reacciones falsas. Están provocadas por la presencia de anticuerpos distintos a los que reaccionan contra el virus del SIDA. Los australianos están convencidos de ello y plantean además otra cuestión. ¿Cómo determinar con exactitud si un paciente cuyas pruebas han sido positivas está realmente infectado por el VIH y no por otra cosa?
Ahora sabemos que los homosexuales, los consumidores de drogas y los hemofílicos, los grupos de riesgo más importantes, al estar expuestos a agentes externos, tienen más capacidad para desarrollar diversos tipos de anticuerpos. En estos casos, es imprescindible conocer la especificidad del test. Es muy posible que este test no indique la infección del virus VIH en ninguna de estas personas.
Lo que significa es que hay una falta de uniformidad en la interpretación de los test, una falta de uniformidad en la comprensión de la naturaleza del test y una falta de uniformidad en la apreciación de los efectos de la enfermedad. Hasta que no se solucionen estos problemas, no debemos considerar el mecanismo del virus como el causante del SIDA.
You talked about oxidative stress earlier. Is treating oxidative stress one of the best ways to deal with the African AIDS epidemic?
I think this is one way to approach, to decrease the rate of transmission, because I believe HIV, we can be exposed to HIV many times without being chronically infected. Our immune system could get rid of the virus within a few weeks if you have a good immune system. And this is the problem also of African people. Their nutrition is not very equilibrated. They are in oxidative stress, even they are not infected with HIV. So their immune system doesn't work well already. So it's prone, it can, allow HIV to get in and persist. So there are many ways which are not the vaccine, the magic name, the vaccine. Many ways to decrease the transmission just by simple measures of nutrition, giving antioxidants, proper antioxidants, hygiene measures, fighting the other infections. So they are not spectacular, but they could, decrease very well the epidemic. to the level they are in Occidental countries,
So,
Western countries.
if you have a good immune system, then your body can naturally get rid of HIV.
Yes.
Oh, interesting. Do you think we should have more of a push for antioxidants and things of that nature in Africa than antiretrovirals?
We should push for more, a combination of measures, antioxidant, but nutrition, advice and nutrition, fighting other infections, malaria, tuberculosis, parasitosis. Most children have worms. Education, of course, genital hygiene for women and men also. Very simple measure, which not very expensive, but which could do a lot. And this is my worry about the many spectacular action. Of course, there are global funds to buy drugs and so on, Bill Gates and so on, for the vaccine. But those kind of measures are not very well funded, not funded at all, or they are, it really depends on the local government to take conscious of this, but local government, they take advice of the scientific advisors from the international institutions, and they don't get this kind of advice very often.
There's no money in nutrition, right? There's no profit.
There's no profit, yes. The water is important. Water is key.
Now, one thing you said, you were talking about the fact that if you have a built immune system, it is possible to get rid of HIV naturally. If you take a poor African who's been infected and you build up their immune system, is it possible for them to also naturally get rid of it?
I would think so.
Okay, that's an important.
It's
point.
important knowledge which is completely neglected. People always think of drugs and vaccine. So this is a message which may be different from what you heard before, no?
The closing?
No. Yes, my message is different from what you heard from Fauci or.
Yes. Yeah. It's a little different.
It's a little different.
Some people are very fortunate that they don't have these side effects.
The number of cases reported went up epidemically, exponentially, because the number of tests that was done went up exponentially. There's some of them that aren't that stupid, but there's a lot of them that are religiously associated with the notion. They believe in the notion that it is an infectious thing that will eventually sweep across the planet and kill everybody.
California microbiologist Dr. Kary Mullis was awarded the Nobel Prize in 1993 for his development of one of the most widely used current techniques in molecular biology today, the PCR, or polymerase chain reaction. This revolutionary technique allows scientists to take a sample containing a very minute amount of DNA and replicate that DNA sequence until there are many copies. One of the first applications of PCR was to analyze human tissue for the presence of HIV. PCR is commonly used in medical and biological research labs for the detection of virus culture in infectious persons.
The people that are AIDS researchers now are getting neurotic if you ask them any questions. There was a time when I first started asking questions, all I wanted was, where are the papers? Just tell me the papers that you read that convinced you that HIV was the cause of AIDS, because I need to reference those papers in some of those. I was working on a test. The first time I really questioned it, I was working on a project where we were measuring HIV in people's blood at this place called Specialty Laboratories in Santa Monica. I was just a consultant there. I came in about 3 days a month, and we were working on that, and at some point we needed to re-up our grant from the NIH to work on that, and I had to write it. And so the first line of that was, HIV is the probable cause of AIDS. And I wrote that, and then I said, well, I need a paper, some kind of scientific paper, to reference that statement, because when you make a statement like that, that's like a fact. You need to say, here's how come I know that, right? You put a little 1 if it's the first statement you've made, and then you put down at the bottom of the paper you have a 1, and you say, here's a paper by somebody that describes why that statement is true, right? And so I said, well, what's that? I don't know. Let me think about, what is that paper? Who do I go to for that? And I looked around. I asked a couple of virologists at that company, and they said, no, you don't have to reference. I said, I have to reference that because I don't know where that came from. How do I know that? And it turned out that nobody knew it. There wasn't a scientific reference, like a paper that somebody had submitted with like experimental data in it and like logical discussion and said, here's how come we know that HIV is the probable cause of AIDS. There was nothing out there like that. Nothing.
Can you tell about your experience when you met Luc Montagnier for the first time and you questioned him about his, whether he had a reference for HIV, since he is the one who...
By the time I met Luc Montagnier, I had met a lot of AIDS researchers at meetings, and I had always gone up to them, if they talked like they knew about HIV and AIDS, I always went up to them afterwards and I said, where can I find a scientific reference that I can use for my, remember I said I had a sentence there, it said HIV is the probable cause of AIDS, and I needed to have that backed up by something before I could write it and submit it. And I went around, I asked a whole lot of people. I said, well, the people, you know, I can't find it. First I looked for it, you know, just in like computer searching kind of stuff like that. But then I said, there's got to be somebody that knows this. You go to experts and ask them. And so I asked all these people one after the other, and none of them had it. None of them. And I was getting really freaked about that. That's when I first started saying, they don't know. Nobody really knows. This whole thing is a big sham. It's ridiculous. But then finally Montagnier came to there was a special little seminar down in San Diego where an old friend of Robert Gallo's, Flossie Wong-Stahl, was opening up a Department of AIDS Research down at San Diego. They had lots of money involved, federal money, and they had Montagnier come there and give a talk. And after that, they had a little wine and cheese thing, and I went over to Montagnier afterwards and I said, Dr. Montagnier, I can't find a reference. Like I can't find a reference to go with the statement, HIV is the probable cause of AIDS. I'm sure you can help me. And he knew that he probably should be able to help me. And he said, well, why don't you quote this new work, and by new he meant like something that came out this year, right? This new work about a virus that can kill monkeys. Or I think it was not monkeys, it was like something related to monkeys, some kind of a baby, a little ape. And I had read that and I said that didn't, it was like supposedly going to be a model system for studying AIDS, that somebody had figured out some kind of retrovirus that passing it back and forth between various mammals, they could finally put it into chimpanzees and kill them. And it killed them in about a week, and it didn't kill them in any, there was nothing like AIDS there. You know, it doesn't kill you in a week. It was just totally ridiculous. None of the symptoms were the same. And I said, well, you know, I read that paper, and I didn't see any connection between that and AIDS, and I don't think that would be a real. I wouldn't want to use that as a reference. And I don't remember exactly what he said, but I know he walked away. Oh, no, before he told me about that paper, he said, why don't you use the NIH, like the CDC report? And I said, well, I looked at that, and that was not a scientific paper. And then he said, what about this other thing, this like paper that had just come out about a month before, and a lot of fanfare associated with that paper, but it was total crap. It was like, yeah, if you get $2 million, you can figure out how to kill a primate with a retrovirus. So what? Doesn't have anything to do with AIDS. It didn't look like AIDS, it didn't smell like AIDS, it wasn't AIDS. It was just like, got a retrovirus that can kill a chimpanzee, so what? So I didn't get any more out of him. He walked away after that. And the people standing around, by the way, who were his colleagues there, looked at him like they were thinking he should come up with a better answer than that. But he couldn't, and he just turned around and walked away. I really thought he'd have an answer. I really did. I mean, that was my last, I was right at the edge of my faith in the system, but I thought Montagnier will know why he thinks HIV causes it, and he'll tell me. He'll say, because of this study, you know, but he didn't have that. None of those guys have that, and that's why they're so weird, you know. That's why they don't want dissent. They don't want people like me walking up and asking them those kind of questions, and they're willing to like go to great lengths to prevent that. They're out on a limb. I wouldn't want to be there with them.
I was given the mission to develop an HIV diagnostic test, and it was sort of an unspoken tradition at Amgen that whenever we would launch into a product development effort, that we'd invite persons from both sides of an issue. And the only person that I knew was not entirely convinced that HIV was an important target was Dr. Peter Duesberg. So I called Dr. Duesberg to come out and give a seminar at Amgen, and he agreed. And it was at that time when he had introduced his theories about other causes of AIDS and his beliefs that HIV was a harmless passenger virus. So that really is what planted the seeds in my life to start questioning the link between HIV and AIDS. And it was interesting at that seminar, the majority of the PhDs in the company actually boycotted the seminar. They had refused to come and be in the same room with Peter Duesberg. And I thought this was odd because as a scientist, we generally love vigorous arguments. And I thought that if persons had arguments against Peter Duesberg, that this would be a perfect time for them to attack him. They did, however, let the research associates come to the seminar, and it was just amazing. Everything that Dr. Duesberg had brought to my attention was completely unknown to me. And based on what he said, I was convinced that there were some serious problems with the HIV/AIDS hypothesis, if indeed what he said was true. And that prompted me to go to the library. And that was about 12 years ago, and I've been studying this issue ever since.
I remember when I made the shift myself, because for the first few years of AIDS, I was reporting on this story from the conventional perspective. And then I learned, from one reason or another, I had a chance to go into it more deeply. I spent a week in the lab of a virologist in Germany who showed me the procedures that were involved in isolating and purifying viruses, showed me why he also didn't believe that this had ever been done with HIV. And gradually I understood the intricacies of this story, and that allowed me to make the shift. But I remember being resistant to thinking that I'd been reporting this wrongly all those years. It takes a lot of, you know, kind of movement inside to be able to change direction on such an important and emotive issue as the cause of AIDS.
From the very beginning of my medical engagement, I focused on family planning, sexual education, contraception, abortion, because I think this is the very basis of a happy life, to be able to control fertility. And in the 87, 88, we were confronted with reports that this new virus and this new disease was breaking out of the formerly so-called risk groups of gay people and hemophiliacs, and was now spreading among the heterosexual population. And I found that highly unlikely and in contradiction to everything I had learned in medicine, because the mode of transmission is predetermined and doesn't change easily. And especially to say, well, now HIV broke out of the risk group because the people inside the risk groups are almost all infected, suggests that the virus has some knowledge about how many people are infected, and realizes and thinks and says, well, now I'm going to change my mode of transmission. This is like us being stuck in a car jam, in a traffic jam, and deciding suddenly, well, now we can just fly because we can't move forward with our car. It doesn't make sense. And that's when I started to ask critical questions. And I didn't get satisfying answers, so I asked even more critical questions. And in the end, I ended up to write a book, Do We Love Dangerously?, to evaluate and bring together and analyze facts about and data available on this question.
Here we have a bunch of people that are definitely sick from some reason. It's likely that their behavior was so radically different than the behavior that had gone. It was an experimental kind of behavior in a way. It's not unlikely that they would have some kind of problems, some health problems. People stop sleeping and eating. People start using all kinds of substances instead of food. And they're associating with it. The world's getting more and more densely populated, and we're spreading more and more diseases around. It's not totally shocking that those people should come down with some diseases that'll kill them. So we don't need to postulate that there was an infection going on, since nobody actually did show that there was. Here comes Gallo and Montagnier and the NIH saying, that's right, it's something else. It's something you didn't have. You're not responsible for it. You're just the beginning of it. We're all going to get it. And they liked that so much, because they said, we can go right ahead and do what we're doing. We just have to do a few little things differently, and then we'll be okay. They cling to that, you know, and they think if you suggest that it's their activity, that the way they live their life, that you're a homophobe or something, you're some kind of an idiot. There's something wrong with you. There's some of them that aren't that stupid, but you know, there's a lot of them that are religiously associated with the notion. They believe in the notion that it is an infectious thing that will eventually sweep across the planet and kill everybody. PCR came along right about the same time that HIV did, and it was at CEDIS that people started looking with PCR for HIV. That was the only way to see it, except for culture, which was a long, protracted procedure, which a lot of times didn't turn up the results there weren't very good either.
Can be contaminated.
Oh, the cultures, the whole method, that cell biology is a bunch of magic half the time. And those culture, you know, the people that say they can do quantitative estimations of HIV from cultures are just, they're fooling themselves. The number of cases reported went up epidemically, you know, exponentially, because the number of tests that was done went up exponentially. How many doctors knew about HIV in 1983? 2. How many knew about it in 1985? Say 500. How many knew about it in 1986? 40,000. So that's where the curve came from. How much money did we make off of HIV this year? And they could have plotted that, and it would have looked the same. You know, and they could have said it's an epidemic because we're making more and more money off of it every year. If it's just caused by needles, or it's just caused by homosexual activities, you're not going to really get a long, sustained public outcry against it, and nobody's going to want to spend $6 billion a year. They're going to say, well, we really didn't like those people anyhow. Great. I can't think of a better solution to the homosexual problem than a disease that'll kill them all. I mean, there would be congressmen that would talk about that quietly, not on television. So the CDC had to say, we can't say that. We got to say it's got to be heterosexually transmitted. There's no proof that it's transmitted at all at that point. So why not just say, well, it's heterosexually transmitted too? Because that made it a plague, and the CDC needed one. The CDC hadn't had a good plague since polio. Their funding was probably going to be cut back if they didn't come up with one. The guy that was the head of the CDC, in fact, wrote memos that have been obtained, you know, where he describes this as hot stuff. You know, those guys have got an agenda, which is not what we would like them to have, being that we pay for them to take care of our health in some way. The scientists are, like you say, they are considered the final arbiters of what's good for the planet or what's bad for the planet. And they hadn't got the slightest idea. Instead of wearing white robes, they wear white lab coats, you know. Instead of bringing you the word of God, they bring you the word of the EPA or whatever. And they don't have to understand for what it is that they're making you do, in fact. And people, you know, just, I think they fall naturally into it because there is a need in humanity for something like a religion, something that makes you feel a part of some larger kind of group, something that you think, in spite of your wormy little life, makes you a part of something good, something big. When beliefs get tangled up with facts, matters of fact become matters of religious kind of significance, where if somebody gets mad at you for asking him a question in a scientific meeting, or says, we're never going to have Kary Mullis at the European Union of whatever kind of scientists, so they put it in Nature, because he came in there and had the audacity to tell people that it might not be that bad to have sex, that AIDS might not be something you get from sex, that I don't see any evidence for it, is all I said. I don't see any evidence for it. Why? You can't tell young people that.
Today there are many scientists and physicians working within the conventional institutions who have similar viewpoints to those of the dissidents. Well, why don't they come out? Why don't they speak their minds? Is it possible that they take a look at what has happened when you do question the HIV equals AIDS equals death hypothesis? They've been suppressed. Dr. Peter Duesberg, noted scientist, was the main person in the line of fire.
Peter is a very scholarly person in addition to being brilliant too. He's very careful. He doesn't say anything that he can't support. And also one of the most brilliant people surely the most brilliant virologist.
Peter Duesberg at the University of California, Berkeley campus is seen as someone who is now dangerous. His funding has been cut off. Graduate students are encouraged not to take his courses, and I think his administrative duties have included being in charge of the department picnic.
My funding, for example, from the National Institute of Health will not be renewed as of this year, and I may be out of business then. Also, my colleagues have essentially excommunicated me. I'm not invited to meetings, or hardly ever to meetings anymore, not ever to one on AIDS. I'm not allowed to publish in the same journals anymore as I used to. Even as a member of the National Academy, I cannot publish in the National Academy my views that drugs may be the cause of AIDS. And I have plenty of evidence to support it, to document my claim, which might be very relevant to the health of millions of Americans and people on this planet. Yet I'm censored because I'm essentially considered to be an outcast or an odd man or somebody who has controversial views.
He was a really well thought of guy too because of his ideas, the whole field. I mean, in cancer, he's the guy that led him into the whole oncogene thing, you know. Big research bonanza. Anybody who could order a little kit from some company and hire a couple of technicians could be a cancer researcher because of things that Peter opened up. And then Peter said, no, I don't think that's actually the cause of cancer. And that made those guys a little bit angry. And then he comes along and does the same thing for AIDS. He said, you know, I think we're on the wrong track here. You know, it's just like in political scandals. Follow the money trail. Figure out who's getting paid for this. Who's getting the money for those Western blots. There's your person who's going to always come down on the side of, yeah, you got a confirmatory Western blots, they call it. They don't even do them in England anymore.
No,
but
not since 1992.
It's totally, and ask a doctor how it works. The doctor who prescribes, it says, got to have a Western blot to confirm this ELISA positive thing. How does that work, doctor? Sir, what are they now measuring about me that's different from what they measured with the ELISA test? He wouldn't know. He's not got any idea. I'll bet you there's scarcely 50 physicians in this country that know what a Western blot really is. They know when to order it, and they know they get a kickback on it probably.
AZT is an antiretroviral drug that became the first drug approved for the treatment of AIDS and HIV infection in 1987. It was originally intended to treat cancer, but it failed to show efficacy and had unacceptably high side effect profiles. However, the unavailability at that time for alternatives to treat AIDS affected the risk-benefit ratio greatly.
The antiviral drugs are chemotherapies. They're all based on chemotherapies that have been developed 30 years ago long before AIDS was known, to kill human cells. Chemotherapy is restricted to a few months in the hope the cancer dies before you die.
If you started taking any other chemotherapeutic agent for the rest of your life, it would be that agent probably that killed you. You know, when you give chemotherapy to somebody with cancer, you give them a round of it for maybe 14 days or a few days. Hopefully you're not going to kill the patient, you're going to kill the cancer. The patient's going to survive. But you don't keep giving it to him until he dies, because he certainly will. The way to get rid of AIDS is to stop funding it. Just stop everything that's called AIDS research. Somebody tries to get a grant for AIDS research, say, we don't do it anymore. We don't have AIDS. If you want to study pneumocystis carinii, we'll maybe look at your paper and see if you've got, you know, something to say about that. You want to study any one of those diseases by itself, try to cure it, we'll talk about that. The thing that I learned, like back in 1968 when I first published a paper by myself in Nature in a field that I had no expertise in at all, there are no old wise men up there at the top of science. Which I really did until 68. I would have thought, you know, if you try to publish a dumb paper in a journal like Nature, it won't get published. If you try to publish a good paper in there, like I later tried to publish PCR, the invention of PCR, in the same journal, and they didn't take it. So it's up there. There isn't an up there. There's no place up on the Academy of Science is just a bunch of idiots, just like everybody else. You know, the editors of journals, austere journals even, they're just busy with their little lives and stuff. There are no old wise men up on the top making sure that we don't do something really dumb.
Campus de Berkeley, California. Aquí, como en otros lugares, hace ya más de 10 años que empezó la lucha contra el SIDA. Pero a pesar de los enormes recursos empleados en esta batalla, la investigación atraviesa por un momento de duda. Algunos científicos han decidido cuestionarse todo de nuevo, empezando por lo que aparentemente se tenía como certeza inamovible: la responsabilidad del virus en el desarrollo del SIDA. ¿Y si los científicos se hubieran equivocado al señalar este virus como agente causante del nuevo síndrome? El mero enunciado de esta crucial pregunta hubiese sido impensable hasta ahora. Sin embargo, escuchen las razones que han conducido a estos científicos a formulárselas seriamente. Son el profesor de biología molecular Peter Duisberg, de la Universidad de Berkeley, y su colega Richard Stroman. Por haber dicho lo que ustedes van a oír ahora, estos dos investigadores han sido tachados de inconscientes. Sin embargo, forman parte de la élite científica norteamericana.
Yo no creo que se haya descubierto aún la causa del SIDA. Desde luego que no. Si se hubiera descubierto, a lo largo de estos 10 últimos años hubiéramos avanzado enormemente en su tratamiento y en su prevención. Pero no ha ocurrido así.
Se están descubriendo muchas cosas acerca del VIH. Esto no se cuestiona. Quiero decir que sabemos más del virus de lo que aparentemente necesitamos saber, pero no estamos descubriendo nada sobre el SIDA.
SIDA, la duda.
Abril 1984. El profesor Gallo anuncia que se ha conseguido identificar la causa del SIDA. La clase científica americana se congratula de esta gran victoria, hasta que se dan cuenta de que el virus, teóricamente encontrado en su laboratorio, proviene de otro lugar. Es aquí, en el Instituto Pasteur de París, donde el virus del SIDA ha sido identificado y aislado por primera vez por el equipo del profesor Montagnier. Terminada la polémica sobre el origen del virus, que mantuvo enfrentados durante un tiempo a Gallo y Montagnier, ambos científicos se ponen rápidamente de acuerdo en identificarlo como el causante del SIDA.
Bien, yo creo que hay que ser cauteloso en este tema. Me parece que lo prudente es que mantengamos la duda. La duda es un hecho científico. No hay dogmas en la ciencia. De cualquier manera, yo no creo que al decir que el VIH es la causa del SIDA esté enunciando un dogma religioso. Mi opinión es que se trata de una afirmación basada en datos. Datos que actualmente son numerosos y que a medida que pasa el tiempo van en aumento. Cada vez es más pequeño el número de personas que rechaza estos datos. Hay que estar ciego, sordo y mudo para no aceptar que este virus está efectivamente asociado al SIDA.
Al igual que el profesor Wayne Hobson que vemos aquí, todos los científicos del mundo coinciden en que el enemigo a combatir es el VIH. Todos menos uno. Peter Duisberg se opone con argumentos a la hipótesis oficial de que el VIH es la causa del SIDA.
Entiendo que Duisberg planteara este tema en su momento, porque entonces aún no existía una toma de conciencia general de que todavía no disponíamos de los elementos suficientes para afirmar que esto era así. Pero yo nunca estuve de acuerdo con Duisberg. Para mí los hemofílicos y los niños infectados eran prueba suficiente. Además estaba la cuestión del SIDA en los monos. Pero él planteó el asunto y una de dos. O le respondíamos o le ignorábamos. La actitud de la comunidad científica despreció en cierto modo a Duisberg porque, en vez de responderle con argumentos científicos, reaccionó escandalizándose. ¿Pero esto qué es? ¿Cómo se puede decir una cosa así en nuestros tiempos?
A partir de 1987, la polémica planteada por Duisberg sobre la implicación del VIH en el SIDA se debatió en la prensa científica. Pero las escasas respuestas aportadas por sus colegas no le hicieron cambiar de opinión. Aún hoy, él opina que no está suficientemente probada la implicación del VIH en el SIDA. El VIH que vemos en estas imágenes pertenece a un determinado tipo de virus, los llamados retrovirus, que Duisberg lleva estudiando desde hace más de 30 años. Él fue el primero que consiguió identificar y descodificar su estructura genética. En 1986, la Academia de las Ciencias reconoció la aportación de sus investigaciones. Desde entonces, Duisberg está considerado como uno de los más importantes especialistas en los retrovirus.
He descubierto que los retrovirus no matan las células, ni provocan enfermedades, a no ser que sean extremadamente activos y numerosos en un animal, y probablemente también en el ser humano, aunque todavía en este último caso no lo hayamos comprobado. Pero aún no hemos encontrado un solo animal o ser humano que esté libre de la presencia de retrovirus. Todos los seres vivos albergan retrovirus idénticos al VIH. Están en estado latente, presentan la misma estructura genética, el mismo mecanismo de replicación, la misma tasa de mutación, idéntica transcriptasa inversa. Son exactamente iguales, pero no producen enfermedades. Son los virus menos dañinos que hemos observado jamás. Por eso, desde hace casi 20 años, muchos de nosotros estamos estudiando los retrovirus como posibles agentes productores del cáncer, precisamente porque no matan a las células.
Duisberg aplica esto mismo al VIH y deduce que el virus del SIDA no puede destruir los linfocitos T4, que son las células que permiten al organismo luchar contra las infecciones. Aun suponiendo que pudiera destruirlas, se ha podido observar que el VIH infecta muy poco los linfocitos T4. Dicho de otra forma, existen muy pocas probabilidades de que pueda provocar daños importantes en el sistema inmunológico. Además, entre el momento de la infección por el virus del SIDA y la aparición de los primeros síntomas de la enfermedad, transcurre un periodo de tiempo que puede llegar a ser de 5 a 10 años o incluso más. lo que demuestra que es un virus de comportamiento absolutamente atípico.
Las enfermedades causadas por virus aparecen cuando muchos virus atacan a muchas células. Si un virus ataca a una sola célula, la infección no es perceptible. Si una persona está infectada por la gripe, la sífilis, la gonorrea o un herpes, al cabo de 2 o 3 semanas aparecen los síntomas. O bien no se presentan nunca porque puede que el sistema inmunológico los reconozca y frene la infección. Por eso pasamos por un periodo de latencia de 1, 2 o 3 semanas, a veces solo de días, porque el virus necesita reproducirse en gran número en nuestro organismo para matar células. Entonces es cuando nos ponemos enfermos. No hay ninguna explicación lógica para aceptar el hecho de que una persona que se infecta hoy consiga neutralizar el virus con su sistema inmunológico y se convierta en seropositiva y desarrolle la enfermedad 10 años después. El virus entra en acción antes de que lo haga el sistema inmunológico. Si el virus fuera el responsable, la enfermedad aparecería mucho antes, no 10 años después.
El último argumento de Duisberg se basa en la presencia de los anticuerpos en la sangre. Según él, deben considerarse como los restos de la batalla entre el organismo y el VIH. La prueba de que el virus ha sido vencido. Para Duisberg no hay ninguna duda. El virus es inocente.
No hay ningún precedente, ni uno solo en la historia de la microbiología, de que un microbio inactivo, latente o no detectable, origine una enfermedad.
¿No se ha encontrado ningún virus activo?
En la mayoría de los pacientes afectados por el SIDA, ninguno. Solamente anticuerpos. Esa es la razón por la cual algunos microbiólogos tan renombrados como Gallo y Robin Weiss no hayan conseguido aislar el virus del SIDA en los pacientes.
¿Y en los nódulos linfáticos?
Tampoco. Lo han intentado durante varios años, pero lo único que han conseguido es el virus de Montagnier. No lo han encontrado porque no hay nada que encontrar.
Está claro. Tras 10 años de polémica, Duisberg mantiene su postura. Según él, todavía no hay nada que pruebe la relación entre el VIH y el SIDA. Y mientras que no se produzca una demostración científica, no cambiará de opinión. La actitud de Duisberg ha llevado a otros investigadores a replantearse el protagonismo del virus en la enfermedad. Como en el caso del profesor Harry Ruben, pionero también del análisis de los retrovirus. Muchos de los especialistas más importantes de hoy en día han sido alumnos suyos.
Solo me gustaría apuntar que, a diferencia de lo que ocurre con otros virus, en el caso del VIH no hay una evidencia que permita su identificación exacta. No descarto que pueda tener cierto protagonismo, pero esto no significa necesariamente que sea la principal o la única causa existente. En cualquier caso, no ha sido probado científicamente.
Las dudas planteadas por los heterodoxos del SIDA no siempre han quedado sin respuesta. Sin embargo, el debate ha sobrepasado el ámbito estrictamente científico y ha llegado a convertirse en una polémica casi religiosa, anatemizadora para los que no participan de las tesis oficiales. Los disidentes excluidos del debate científico se han agrupado en torno al profesor de bioquímica Charles Thomas, con la intención de hacerse oír.
La opinión generalizada entre la gente es que el causante del SIDA es un retrovirus conocido por VIH. Sin embargo, muchos científicos biomédicos ponen en duda esta hipótesis. Nosotros proponemos que un grupo de científicos independientes pondere de manera exhaustiva todas las pruebas existentes a favor y en contra de esta posibilidad. También proponemos que se efectúen estudios epidemiológicos críticos. Hemos enviado nuestra propuesta a varias publicaciones científicas, pero ninguna la ha publicado, a pesar de que iba avalada por la firma de casi un centenar de prestigiosos investigadores.
Hasta el momento, 3 premios Nobel han manifestado públicamente sus dudas acerca de la tesis oficial. La bióloga Barbara McClintock, ya fallecida, el profesor Walter Gilbert, de Harvard, y por último, Kary Mullis, Premio Nobel de Química en 1993, a quien vemos en la imagen. Actualmente reside en San Diego, en la costa de California. Kary Mullis es el inventor del APCR, un procedimiento que permite aislar y observar los genes ampliados de un microorganismo. Este descubrimiento ha revolucionado el trabajo de los científicos en todos los laboratorios del mundo y se ha convertido en un instrumento valiosísimo en la investigación sobre el SIDA, lo que ha llevado a Mullis a profundizar en este tema y a poner en duda determinados aspectos incuestionados hasta ahora.
Yo intentaba encontrar unas referencias, es decir, los trabajos científicos que demostraran que el virus VIH era la causa del SIDA. Necesitaba estos datos porque estaba redactando un informe y tenía que citar estas referencias para poder decir, esto es así, como confirman tales y tales documentos. Pero no conseguía encontrarlos. En anteriores trabajos nunca había tenido el menor problema a la hora de buscar las fuentes científicas sobre las causas de algún fenómeno. Normalmente es fácil encontrar estos datos recurriendo a publicaciones especializadas. Se averigua quién ha estudiado determinado tema, qué descubrimientos ha realizado y cuál es su tesis de trabajo. Pero esta vez no fue así. Pregunté a todos los investigadores sobre el SIDA por qué opinaban que el VIH era la posible causa de la enfermedad. ¿Cómo tenían la certeza? ¿Con qué documentos trabajaban? Todos me contestaban, pues hay un montón de datos que lo avalan. Y yo les decía, ¿podría decirme exactamente cuáles? Yo estaba deseando encontrar lo que buscaba para terminar mi trabajo. Necesitaba esos datos para citarlos en mi informe sobre el VIH en la sangre. Solo quería eso. Solo quería documentar mis teorías y para ello necesitaba la prueba de que el VIH era la posible causa del SIDA. Pero no pude conseguirlo. Nadie me daba la respuesta. Siempre me contestaban que los papeles los tenían en la oficina y yo decía, bueno, pues envíeme esos documentos desde su despacho, pero nadie lo hizo.
Para Mullis, esta dificultad en conseguir las pruebas demuestra que la investigación se ha realizado sin respetar las reglas más elementales de la metodología científica. Mullis es de la opinión de que en el tema del SIDA todo se ha hecho con demasiada precipitación. La investigación, la divulgación pública, el intercambio de opiniones entre la comunidad científica y, por último, la conclusión a la que se ha llegado, que no es más que una verdad a medias.
Normalmente la ciencia no suele discurrir por esos caminos. Cuando se quiere afirmar algo hay que experimentarlo previamente para obtener unos resultados. Hay que estudiar el tema con detenimiento y preguntarse si hay algo en lo que nos hayamos podido equivocar. Si es así, conviene realizar un nuevo experimento para comprobar los datos. Las ruedas de prensa deben convocarse después de concluir este proceso. Esta es la manera de establecer las evidencias científicas. En el caso del SIDA, efectivamente se convocó una conferencia de prensa en la que se afirmó: el VIH es la causa del SIDA. El Centro de Control de Enfermedades en Atlanta se hizo eco de estas palabras añadiendo: cuidado, el VIH es un organismo peligroso. Si lo encuentran en un paciente que presenta determinado tipo de síntomas, deben informarnos. Y se lo comunicaron a todos los médicos. Sin embargo, no explicaron por qué lo sabían. Se convirtió en una verdad con demasiados interrogantes, pero no puede considerarse en modo alguno una verdad científica.
Mullis opina que se han realizado suficientes estudios sobre el SIDA. Sin embargo, aún no tenemos los datos necesarios para confirmar que el VIH es su agente causante. Necesitamos una evidencia científica que demuestre la certeza de esta hipótesis.
No disponemos de una evidencia científica, solo hay un muestrario de argumentos. Permítanme un símil. Tenemos un culpable al que no hemos visto asesinar a su víctima. No le han pillado infragante, pero existen pruebas en su contra, manchas de sangre o lo que sea, y se le lleva a juicio. Y ocurre como en el caso del señor O.J. Simpson, que aunque hay ciertas evidencias que le inculpan, por distintos motivos el asunto toma un sesgo diferente.
El profesor Montagnier dispone de un descubrimiento reciente que contradice los argumentos de sus adversarios sobre el estado latente del virus previo al desarrollo de la enfermedad. Dos artículos publicados en la revista científica Nature sostienen la teoría de que el virus no pasa nunca por un periodo latente. Desde que el individuo se infecta, el VIH y el sistema inmunológico libran una batalla encarnizada. Batalla que finaliza con la victoria del virus. John Maddox, redactor jefe de la revista, ha pedido a Duisberg, a Mullis y a los que rechazan el protagonismo del VIH en el SIDA que reconozcan públicamente su equivocación.
Si Maddox piensa que esos artículos prueban que el virus VIH provoca el SIDA, debería pegarse un tiro. Nunca ha aceptado las hipótesis alternativas de las personas como yo. Siempre ha mantenido una opinión cerrada. ¿Por qué todo el mundo ha aceptado sin pestañear que este virus es el causante? Si hasta 1995 no se sabía el origen del SIDA, ¿por qué llevan 10 años actuando como si lo hubieran sabido desde el principio?
A pesar de los artículos de Nature, Mullis mantiene su postura no solamente por lo que acaba de explicar, sino fundamentalmente porque estos trabajos no aportan, según su opinión, ninguna luz sobre el papel del virus en la destrucción del sistema inmunológico. Lo que, proviniendo de un premio Nobel, constituye una razón de peso.
Estoy absolutamente convencido de que no es la causa del SIDA. Los hechos están ahí. Si queremos tenerlos en cuenta, demostrarán que el VIH no es el causante del SIDA.
¿Existe el SIDA?
Todas las enfermedades que se asocian al SIDA existen. Muchas de ellas desde hace mucho tiempo. Pero nunca aparecían en las mismas personas a la vez, lo que parece indicar la presencia de algún tipo de síndrome. En realidad, si analizamos dolencias como la neumocistis carinea, el sarcoma de Kaposi y la aparición de hongos en la garganta, en la cabeza, en la cara y en las extremidades, descubriremos que todas esas afecciones tienen una causa determinada, pero que no siempre es la misma. No hay ninguna razón para creer que todas responden a la misma causa.
El sarcoma de Kaposi. A principios de los años 80, cuando aparecieron los primeros casos de SIDA en San Francisco, los homosexuales infectados presentaban un tipo de cáncer de piel muy poco común, el sarcoma de Kaposi. Esta enfermedad se manifiesta por la proliferación de manchas marrones por todo el cuerpo. Para la mayoría de los especialistas, el sarcoma de Kaposi en los homosexuales contaminados por el virus se convirtió rápidamente en la marca identificativa del SIDA, su seña de identidad más evidente. Sin embargo, hasta el momento nadie ha podido establecer de forma clara el papel del virus en la aparición del sarcoma de Kaposi. Es más, se ha descubierto un nuevo agente, otro posible culpable. El doctor Harry Habercos del Departamento de Drogas de Rockville, Estados Unidos, ha estudiado los efectos de las drogas. Él ha sido uno de los primeros en establecer una posible relación entre la aparición del sarcoma de Kaposi y el popper, una droga afrodisíaca muy extendida entre los homosexuales hasta los años 90.
Hicimos un estudio entre los gays, analizando su comportamiento, sus hábitos sexuales, las drogas que tomaban, etc. Y descubrimos que los que padecían el sarcoma de Kaposi tenían patrones de comportamiento distintos a los que, por el contrario, desarrollaban neumocistis. Y descubrimos que la diferencia radicaba en la utilización o no del popper.
Ante esta evidencia, el Departamento de Drogas realizó numerosas campañas de prevención, advirtiendo del peligro del uso del popper en grandes dosis. Posteriormente fue prohibida la venta del popper, y el resultado se ha visto reflejado en los últimos estudios epidemiológicos.
En los últimos años se ha producido un notable descenso en la aparición del sarcoma de Kaposi, tanto en términos absolutos como porcentuales.
¿A qué es debido?
Bueno, yo creo que guarda relación con la inhalación de nitrato. Últimamente ha descendido el uso de este producto. Sin embargo, no sabemos si se ha producido una disminución del virus entre estas personas. Tampoco hemos podido probar con exactitud la relación con los herpes. Sin embargo, la epidemiología del sarcoma de Kaposi y la de la inhalación del nitrato en los Estados Unidos parecen íntimamente ligadas.
Para ser exactos, hay que decir que el sarcoma también aparece en pacientes que nunca han utilizado el popper, así como en personas que no están infectadas por el VIH. lo que demuestra que las cosas son mucho más complicadas de lo que se pensaba en un principio.
El agente responsable no está muy claro. Sabemos que estos nuevos herpes han aparecido en algunos afectados y también conocemos su relación con el popper. Puede que el VIH no tenga necesariamente que ver, pero lo que es seguro es que además del popper tiene que haber otro agente responsable.
El origen de la enfermedad sigue siendo incierto. Aunque el avance de las investigaciones apunta a restar protagonismo al virus VIH, en nuestros días el sarcoma de Kaposi sigue estando asociado a las enfermedades propias del SIDA. Nos encontramos en un punto muerto.
Hasta ahora hemos creído en la ecuación VIH igual a SIDA. Pero si el virus no es la causa del sarcoma de Kaposi, nadie debería considerar el SIDA como una enfermedad nueva y tampoco nadie debería creer en el VIH. Y ahora se olvidan de todo, menos del virus. Resulta que hasta el sarcoma de Kaposi no es una enfermedad del SIDA. ¿Por qué no nos olvidamos también de la neumonía? ¿Y entonces qué nos queda? Nada.
Pasemos a otro tema, cambiando de escenario. Recordemos este anuncio publicitario de 1987.
But now we know every one of us could be devastated by it. The fact is, over 50,000 men, women and children now carry the AIDS virus. That in 3 years, nearly 2,000 of us will be dead. That if not stopped, it could kill more Australians than World War II.
Sin embargo, hasta el momento en Australia se han detectado menos de 10.000 casos de SIDA, en vez de los 50.000 que preveía el anuncio. Fundamentalmente homosexuales y toxicómanos. No queremos quitarle importancia a la enfermedad, ni señalar acusadoramente a los grupos de riesgo. Afortunadamente, a veces, las previsiones se equivocan. En Francia, Alemania y otros países europeos, la epidemia se ha estabilizado. Incluso comienzan a aparecer síntomas de receso. No se ha producido la anunciada generalización de la enfermedad entre los heterosexuales. Seguramente han influido las campañas de prevención, pero también existen otras razones.
Las cepas microbianas existentes en Europa no presentan una capacidad de transmisión poderosa entre los heterosexuales, por lo que no se ha producido un contagio masivo. Lo ha habido en cierta medida entre las mujeres, fundamentalmente entre las drogodependientes y las que tenían parejas bisexuales. Pero no ha habido una reacción en cadena. Podemos concluir que no ha existido una transmisión generalizada en la población heterosexual. Y creo que no se producirá. Puedo citar como ejemplo el caso de las prostitutas. Las encuestas demuestran que solamente se han infectado un 2 o un 3% de ellas y que este porcentaje no va en aumento, excepto en el caso de las toxicómanas.
Los estudios epidemiológicos realizados sobre las prostitutas no toxicómanas en las grandes ciudades europeas confirman las conclusiones del profesor Montagnier. De hecho, en algunas ciudades europeas no se han encontrado prostitutas seropositivas. Si partimos de la base lógica de considerar a las prostitutas como el grupo de mayor riesgo entre la población heterosexual, podemos deducir que el contagio por el virus del SIDA entre el resto de los heterosexuales será aún menor. ¿Debemos, por tanto, concluir que no se producirá una propagación del virus entre la población heterosexual? Los datos que posee el Centro Europeo para la Vigilancia del SIDA, organismo que dirige la mayor parte de los estudios epidemiológicos, no apuntan en esa dirección.
El VT2 diferente del VT3.
En general, es verdad que los homosexuales y los toxicómanos siguen siendo, 15 años después de la aparición de la enfermedad, los grupos más afectados. Mucho más que la población heterosexual. Pero esto no quiere decir que en el futuro no se produzca la propagación del SIDA a este otro segmento de la población. En realidad, estamos asistiendo a una difusión lenta y progresiva del virus, mucho menos fulminante que la de los otros dos subgrupos. La extensión de la enfermedad parece haberse estabilizado. Pero Este fenómeno no debe hacernos bajar la guardia. Lo que ocurre es que el riesgo de contagio de los heterosexuales es menor y, por tanto, la epidemia se extiende lenta y dificultosamente.
La amenaza del SIDA que ha planeado sobre Europa en los últimos 10 años parece haberse alejado para centrarse en África. Desde allí, desde el otro lado del Sáhara, nos llegan imágenes espeluznantes que no permiten albergar la menor duda. El SIDA azota a todos por igual: ancianos, jóvenes, mujeres y hombres. Según la Organización Mundial de la Salud, millones de personas están infectadas por el VIH. La epidemia se ceba en el continente. ¿Quién podría poner en duda la magnitud de la tragedia? En estos países, la polémica no se centra sobre el alcance de la enfermedad, sino sobre el nombre que debemos asignarle. África se muere, pero ¿de qué?
Es verdad. Sé por experiencia que los test que se realizan aquí, muy sensibles y específicos, pierden eficacia al aplicarlos a sueros africanos. Ello es debido a varias razones. En primer lugar, los africanos padecen infecciones recurrentes o crónicas, como el paludismo, lo que provoca una tasa de anticuerpos anormalmente elevada. Por tanto, se pueden producir reacciones no específicas que den lugar a errores en los resultados, a falsos seropositivos en las pruebas de SIDA. También existe una enorme diversidad en las cepas africanas. Encontramos allí todas las variantes posibles del virus. Y en muchas ocasiones las pruebas no están convenientemente adaptadas. Por tanto, es necesario realizar un estudio exhaustivo. ¿Ponen en duda nuestras hipótesis los datos que llegan de África? No lo creo, porque todas las estimaciones han sido obtenidas mediante métodos diferentes y muestran un abanico de posibilidades. Basta visitar cualquier hospital de África para darse cuenta de que la mayoría de los pacientes padecen el SIDA.
Partiendo de estos mismos datos, una publicación británica ha expuesto unas conclusiones radicalmente distintas. En octubre de 1993, el Sunday Times publicó un reportaje encabezado por un titular sensacionalista: El mito del SIDA en África. Neville Hodgkinson, su autor, periodista especializado en temas científicos, plantea nuevos interrogantes tras visitar África Central en 1993. En su opinión, hay algo que no encaja en la versión oficial. ¿Está África realmente enferma de SIDA?
No si definimos el SIDA como una nueva enfermedad infecciosa. África sufre y se muere debido a la malnutrición, al resurgimiento de la malaria, a la tuberculosis. Muchas personas necesitan atención médica, pero no, yo no he encontrado ninguna evidencia que demuestre el surgimiento de una nueva epidemia.
Neville Hodgkinson viajó por África durante varias semanas. Primero visitó Kenia, después Tanzania, luego Zambia y por último Zimbabue. Nos cuenta el miedo de los africanos al SIDA, más extendido que la propia enfermedad, y las razones que lo explican.
A mediados de los años 80 llegaron los científicos americanos con los nuevos test del SIDA y empezaron a aplicarlos a grandes grupos de población. Observaron que casi el 50% de las personas estudiadas daban positivo en las pruebas y proclamaron: el VIH está aquí desde hace muchos años. África está a punto de sucumbir a la mayor epidemia de la historia. Y claro, crearon un clima de auténtico pánico.
El terror está presente en todas partes, y de forma infundada, según escribe Neville. Él no ha visto el SIDA. No lo ha encontrado en las aldeas diezmadas por la malaria, ni en los cementerios improvisados para hacer frente a la hecatombe. África se muere, como siempre. a causa de algo que no tiene nada que ver con una nueva enfermedad que en Occidente llamamos SIDA.
Cuando llegué a África, lo primero que me sorprendió fue no encontrar esos hospitales atestados de enfermos de SIDA, esos pueblos arrasados por la enfermedad de los que tanto se había escrito. Pero cuando preguntaba de qué se estaban muriendo, siempre me contestaban, de SIDA. Yo decía, bien, ¿pero de qué enfermedad? Porque el SIDA es un síndrome de inmunodeficiencia. Y me contestaban, bueno, este paciente tiene tuberculosis, este otro una diarrea crónica y este malaria. Había muchas enfermedades diferentes, fiebres, diarreas, todas las que ha habido siempre en África y que ahora los médicos han rebautizado como SIDA, debido al miedo que provocaron los test que se realizaron en su día.
¿Es posible que se haya cometido un error de tal magnitud? Hoy en día muchos investigadores se han planteado la misma pregunta. El profesor Essex, un científico norteamericano, ha realizado recientemente un estudio en Zaire en el que muestra que las personas afectadas por la lepra, no por el VIH, reaccionan positivamente a las pruebas del SIDA. Paradójico. Essex resalta que los resultados de los test deben ser interpretados con suma cautela cuando se trata de pacientes infectados por enfermedades micobacterianas, como por ejemplo la lepra y la tuberculosis. Afirma que los test no son un elemento suficiente para diagnosticar el SIDA en las regiones de África Central donde dichas enfermedades están muy extendidas, es decir, casi todo el continente.
Es una pésima noticia, porque la tuberculosis y la lepra están absolutamente extendidas por toda África. Pero como siempre que informamos sobre esto, ningún científico especializado se ha hecho eco de nuestras palabras. Pretenden seguir adelante como si nada hubiera ocurrido.
Hasta el momento, Hodgkinson no ha obtenido respuesta alguna. Ni por parte de los periodistas que están en desacuerdo con él, ni por parte del propio profesor Essex. ¿Los test del SIDA son menos exactos de lo que se dice? Aquí, en Australia, un equipo de investigadores de Perth ha realizado un estudio exhaustivo sobre este tema. Y precisamente en este hospital. Tres investigadores han revisado prácticamente todo lo que se ha dicho y escrito sobre las pruebas del SIDA para intentar responder a esta pregunta. ¿Un test positivo prueba de forma inequívoca el contagio por el virus del SIDA? El resultado de su trabajo sobre el Western blot, el test teóricamente más fiable de todos los existentes, fue publicado en 1993 en la revista Biotechnology.
Hay muchos datos que demuestran que el test ha sido considerado fiable antes de haber sido convenientemente comprobado. Este es el punto clave de nuestro trabajo. Se ha puesto en tela de juicio la fiabilidad del test. Quizá estemos dando por buenos unos resultados inciertos.
Debemos recordar que el test jamás detecta el propio virus, sino los anticuerpos que han reaccionado a la presencia del virus en el organismo. ¿En qué consiste el test del SIDA? Se desarrolla en dos etapas. La primera es el test ELISA. Si el resultado es positivo, debe ser obligatoriamente confirmado por una segunda prueba, el test de Western blot. El Western blot consiste en una bandeja sobre la que han sido fijadas las diferentes proteínas del virus. Dicha bandeja se pone en contacto con el suero del paciente. Si el suero contiene anticuerpos contra el virus, las proteínas aparecen en forma de bandas negras. El test es positivo cuando la bandeja presenta varias bandas negras. Hasta aquí todos de acuerdo. Pero posteriormente las cosas se complican porque los criterios valorativos cambian de un país a otro. Francia no tiene los mismos criterios que los Estados Unidos para aceptar una prueba positiva. Dichos baremos varían también en Australia o en África.
En estas columnas podemos observar los distintos criterios que se emplean para definir un test Western blot positivo, es decir, para establecer cuándo una persona está o no infectada por el VIH. Los criterios que se aplican varían en cada país e incluso en las distintas instituciones de un mismo país. Si aparecen dos bandas negras en el test de un paciente, sería considerado seropositivo en África y en algunos estados de Norteamérica, pero no lo sería en Australia, ni en otros estados americanos, ni en Francia. Si aparece una tercera banda, una P24, no será considerado seropositivo en Australia, ni en muchas partes de América, ni en Francia. Si aparecen cuatro bandas, tampoco se les considera infectados por el VIH ni en Australia ni en Francia.
No puede considerarse infectada a una persona aquí y allí no. O se está infectado por el virus o no se está. Con estos criterios no vamos a ningún lado.
No es fácil encontrar una explicación lógica a la arbitrariedad de estos baremos. Se deberían aplicar los mismos en todas partes. ¿No sería más sencillo y más seguro? Pero los laboratorios no son de la misma opinión. La sangre contaminada, dicen, sean cuales sean los criterios aplicados, reacciona positivamente y siempre aparecen todas las proteínas del virus. En ello radica la diferencia con la sangre no contaminada.
Aunque se unifiquen los criterios, seguiremos teniendo el problema de la imposibilidad de repetir un mismo test. Es muy difícil hacer dos tests idénticos. Por ejemplo, este test de una misma sangre ha sido realizado por 19 laboratorios norteamericanos diferentes. Los mejores laboratorios, los más prestigiosos.
Cada bandeja corresponde a un laboratorio distinto. En algunas aparecen varias bandas negras, en otras tan solo algunas. En la bandeja del centro, en la letra I, no aparece ninguna. Pero el suero pertenece a un mismo paciente infectado por el virus del SIDA. Los laboratorios australianos han detectado errores aún más graves.
Para intentar explicar el porqué de estas enormes diferencias, me gustaría mostrarles el estudio realizado por los analistas norteamericanos. En 1991, el Diario Americano de Patología Clínica envió 101 muestras negativas, es decir, que no tenían bandas de sueros Western blot, a 3 laboratorios distintos. En 67 muestras contestaron que no aparecían bandas. En 13, que aparecían bandas de distinto tipo. En una, P41 y en otra P24. En 2 muestras P24, 32 y 41-120, lo que para muchos laboratorios se considera un resultado positivo. Sin embargo, todos los sueros eran negativos.
Es decir, que el análisis de un suero puede resultar positivo en un laboratorio y negativo en otro. Los australianos opinan que los resultados contradictorios ponen en entredicho la validez del propio test, su capacidad para determinar si una persona está o no infectada por el virus, lo que los científicos denominan especificidad.
La especificidad de un test asegura que el resultado sea positivo solo cuando el virus está presente en la sangre. Si el virus no está en la sangre y a pesar de ello hay un resultado positivo, el test no es específico.
Los australianos han llegado a afirmar que los resultados positivos en las pruebas del SIDA no confirman de manera absoluta que una persona esté infectada por el VIH. Para comprobarlo, han realizado pruebas de SIDA a ciertos animales que no sufren la infección por el virus. Por ejemplo, a estos ratones, a los que se les ha inyectado sangre de otro ratón y se les han encontrado anticuerpos. También a estos perros. De los 144 perros utilizados, la mitad presentó los mismos resultados que los ratones. Se han realizado pruebas a partir de sueros extraídos en 1968 a indios amazónicos. Muchas de ellas dieron resultados positivos a pesar de que en esas tribus no se ha producido nunca ni un solo caso de SIDA. Lo mismo ocurre entre algunas poblaciones de ciertas regiones de África. Los virólogos que trabajan con el virus del SIDA están acostumbrados a este tipo de reacciones falsas. Están provocadas por la presencia de anticuerpos distintos a los que reaccionan contra el virus del SIDA. Los australianos están convencidos de ello y plantean además otra cuestión. ¿Cómo determinar con exactitud si un paciente cuyas pruebas han sido positivas está realmente infectado por el VIH y no por otra cosa?
Ahora sabemos que los homosexuales, los consumidores de drogas y los hemofílicos, los grupos de riesgo más importantes, al estar expuestos a agentes externos, tienen más capacidad para desarrollar diversos tipos de anticuerpos. En estos casos, es imprescindible conocer la especificidad del test. Es muy posible que este test no indique la infección del virus VIH en ninguna de estas personas.
Lo que significa es que hay una falta de uniformidad en la interpretación de los test, una falta de uniformidad en la comprensión de la naturaleza del test y una falta de uniformidad en la apreciación de los efectos de la enfermedad. Hasta que no se solucionen estos problemas, no debemos considerar el mecanismo del virus como el causante del SIDA.
La mayor parte de los científicos cree que los problemas que surgen en la interpretación de las pruebas son muy escasos. Además, para evitar errores, se realizan numerosos controles de calidad. La duda planteada desde hace ya más de 10 años por los investigadores que aparecen en este reportaje ha comenzado a socavar las verdades oficiales. También ha anidado en el seno de la comunidad científica. En esos círculos, la polémica está sobre el tapete, con menor virulencia ciertamente que en casos anteriores. La ecuación VIH igual a SIDA ha sido revisada a la luz de los nuevos descubrimientos sobre la enfermedad, y este proceso se ha visto reflejado en la prensa.
Todo hubiera sido muy sencillo si, tras descubrir el virus del SIDA, hubiéramos descubierto un medicamento antirretrovírico que frenara la enfermedad y curara al paciente. Pero en realidad nos dimos cuenta de que las cosas eran mucho más complicadas. Una sustancia que in vitro era capaz de frenar la reproducción del virus del SIDA no servía para bloquear la enfermedad en los pacientes. Había que seguir buscando. Había que intentar llegar a comprender por qué la epidemiología del SIDA no es igual en los países industrializados que en los países en vías de desarrollo, en África o en Asia. Por tanto, había que intentar dar respuesta a muchos interrogantes, y la presencia del virus no explicaba por sí misma todos ellos.
El propio Luc Montagnier, descubridor del virus, ha sido uno de los primeros en relativizar el papel del VIH y señalar la existencia de otros factores asociados al virus que desencadenan el SIDA. Nos encontramos en San Francisco en 1990, en un momento muy importante en el que se estaba celebrando la 6ª Conferencia Internacional sobre el SIDA. Durante dicha conferencia, Luc Montagnier afirmó que el VIH podía no ser la única causa del SIDA. Para el virólogo Pasteur, el VIH tiene un cómplice, el micoplasma. Sin este acompañante, el virus no puede actuar. El tiempo no ha dado la razón a Montagnier. Hoy en día el profesor tiene una actitud más flexible, pero en el fondo no ha cambiado de opinión.
Puedo decirle que tenemos argumentos muy sólidos para afirmar que junto a otros factores infecciosos conocidos, los virus y las bacterias, poseen también las propiedades necesarias para desempeñar un papel importante en el SIDA. Ahora se trata de averiguar el papel del micoplasma, su interacción con el virus. ¿Precede a la infección viral y se extiende a causa de la inmunodepresión provocada por el virus? ¿O bien se produce una infección simultánea porque el micoplasma, que probablemente también se transmite por vía sexual, puede contagiarse al mismo tiempo que el virus? ¿Qué papel desempeña en el desarrollo de la enfermedad? Por ahora no tenemos una respuesta. Los últimos descubrimientos realizados en el laboratorio apuntan a la idea de que el micoplasma puede tener un papel muy importante en el desarrollo del SIDA.
Sin embargo, a pesar de las últimas aportaciones, para la comunidad científico-médica, el virus VIH sigue siendo el principal culpable. Algunos científicos, como Duisberg y Rubin, que no comparten las tesis aceptadas por los investigadores de casi todo el mundo, se ven obligados a aventurar nuevas hipótesis. A buscar nuevos culpables, con sus escasos medios y con la única ayuda de la certidumbre basada en la experiencia adquirida en sus campos de investigación.
En todo el país, En todos los pacientes de SIDA, no se ha realizado ni un solo estudio científico sobre los efectos a largo plazo de las drogas inyectadas o inhaladas. No se ha llevado a cabo ninguno. Aunque ya han muerto 150.000 afectados por el SIDA, que eran consumidores de drogas intravenosas. 250 que inhalaban popper, tomaban anfetaminas, y que murieron o enfermaron de SIDA debido al consumo de estas drogas.
Básicamente, mi opinión es que hay muchos factores coadyuvantes y que el VIH puede ser uno de ellos. No descarto esa posibilidad, como tampoco descarto el papel de las drogas y de otros hábitos de vida. Las drogas intravenosas y otros agentes de contagio, e incluso la intervención de otros microorganismos.
Esta hipótesis puede resultar sorprendente, pero cuando menos merece ser científicamente argumentada. Pero esa es otra historia. Nuestro objetivo es demostrar que existen varios puntos oscuros en torno a las causas del SIDA y que, por prudencia, se debería prestar atención a todas las posturas, a todas las hipótesis, hasta las más inquietantes. Nos gustaría que el siguiente testimonio fuera algo más que una simple aportación. Nos referimos a Daniel Kosland, antiguo redactor jefe de la revista Science. Escuchemos sus palabras.
Seguiremos debatiendo sobre la causa del SIDA hasta que consigamos curarlo. Hasta entonces, siempre habrá personas que digan que no vamos por buen camino, que deberíamos intentarlo por otras vías. No creo que consigamos evitar que pongan en tela de juicio nuestro trabajo hasta que encontremos la solución. Cuando ésta aparezca, el debate habrá llegado a su fin.
¿Considera positivo que personas como Duisberg y otros mantengan abierta la polémica?
Sí, absolutamente. Aunque Duisberg ha hecho afirmaciones con las que no está de acuerdo la mayoría de la gente, es importante que se conozcan para demostrar que no somos un búnker apegado a una verdad absoluta. Es el único camino posible.
You speak of the case in Australia, yeah. Well, in Australia there was a man who it's claimed infected 3 women, and I think 1 woman died. And the defense of the man, I don't know the details, if he did it deliberate, if he knew. I don't want to get into that and what the punishment should be. But the question is his defense was an absurdity, but that the virus didn't exist.
The biggest problem with the HIV theory of AIDS is HIV.
There is a group of AIDS denialists that say that HIV does not exist and has never been isolated, which is as bizarre as it gets.
You and your colleagues not only state that HIV does not cause AIDS, but you take an even greater leap and say HIV does not exist. Is that correct?
Oh, it is partly correct. We do not say that HIV doesn't exist. It may exist, but the presently available data does not prove its existence.
But how can you say that when world-renowned scientists like Dr. Gallo and Dr. Fauci say HIV does exist? Are you telling me and the world that they're all wrong?
No, what we're saying is there is the evidence there, the data in the scientific literature, which is published. Scientists interpret data differently, and we interpret the evidence one way, and they interpret it in a different way. So in our view, the evidence does not prove the existence of HIV.
we've all seen pictures. We've seen electron micrographs of HIV. How can you say something that we see isn't there?
You didn't see electron micrograph of HIV. What we see is electron micrograph of particles which look like retroviruses. But it's one thing to look like and another thing is to be a virus.
The one thing I don't understand is how can you question the existence of a virus when there was an international lawsuit against the United States government and Robert Gallo for stealing the French virus? I mean, it seems to me there must be a virus there if somebody stole it.
That's the problem. Under no circumstances Robert Gallo could have stolen Montagnier's virus, even if there was such a virus. Because what Montagnier sent to Gallo was supernatants from his cultures. And in the supernatant, the viruses don't last for too long. And in fact, the particles have to have knobs on their, or spikes on their surface to be infectious. And these knobs are lost. And in fact, nobody has proven that they exist. But even if they exist on HIV, or the particles, they are lost very rapidly. So it is impossible for Montagnier's, for Gallo to have stolen a virus from Montagnier.
So can you prove to me that HIV doesn't exist?
I can show you what the evidence shows, what Montagnier, for example, presented, because everybody accepts that Montagnier is the discoverer of HIV. And I can show you the evidence which Montagnier presented and claims to have proven the existence of HIV. And I will explain to you why Montagnier's evidence does not prove the existence of HIV.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
Now, Dr. Gallo and Dr. Fauci talked a lot about isolation and purification. Can you tell me what the difference is between the two?
Isolation, what was it?
Isolation and purification.
Of the virus?
Yes.
Well, you isolate a virus by finding the virus which causes a disease. You purify a virus by making a lot of, just by purifying it so you get a pure virus.
Okay.
I don't understand what the issue.
They interchanged the two, and I wasn't sure
I see.
If it was the same thing or if it was two totally different.
No, it depends on how they use it.
Okay. Can you explain the process of HIV isolation?
Well, didn't Dr. Gallo do that? He actually isolated it. So, yeah. Why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. Okay. I got other things to do.
Isolation is essentially getting the virus from the patient and being able to transmit this virus to another cell to reproduce the infection and to have a continual supply of the virus. And that's called an isolation. Purification is just obtaining the virus free of cellular contaminants or other contaminations. But it doesn't mean necessarily that the virus is infectious.
Virus isolation, I would take to mean that you take some infected material, like a blood sample from someone who you think or believe may have HIV infection, may already have symptoms of AIDS, and you try and grow the virus from that blood sample. So you would put the blood sample into culture, you would stimulate the lymphocytes to proliferate with various growth factors or cytokines, and you would harvest the virus from the culture medium that you're growing it in. You would spin out the cells separate the cells from the supernatant, and you would look for the virus in the culture medium. That needn't be a pure medium, because you can use markers of the virus that tell you it's there, such as the reverse transcriptase enzyme. So you can see evidence of the virus without actually purifying particles. In that very first paper from the French group, published in May 1983, there were two things that appeared to class it amongst the retrovirus family. One was reverse transcriptase activity, and one was actually looking at virus particles with the electron microscope.
The title of the 1983 paper by Montagnier and his colleague is Isolation of a T lymphotropic retrovirus from a patient at risk of acquired immune deficiency syndrome, AIDS. Now, the word isolation means to obtain something, or to place something apart, or to obtain a substance separate from everything else. Apparently, Montagnier by isolation did not mean this, but something totally different. And I'll explain to what he meant by isolation. He did three main experiments. The first experiment, he took lymphocytes from a patient, now is known as BRU, and he cultured them with many substances, including PHA and other growth factors.
What are growth factors?
They are substances which make cells to multiply.
Okay.
Right? Or to live, and to stay alive in the culture. And after 15 days, he discovered their reverse transcriptase activity. And they claimed that this proved the existence of a retrovirus in their patient, in their patient's cells.
Every 2 days we were taking the supernatant, looking for the presence of the enzymes that transform RNA to DNA, the reverse transcriptase, because we know that this enzyme is associated to virus particles. So we were testing for this enzyme activity in the supernatant.
What's so special about reverse transcription?
Anytime you're searching for a new agent, you want to have some simple measurement of the presence of that agent. In times past, you would put it on cell culture in the laboratory, and suddenly these beautiful cells will all start turning into dead cells. You say, oh, something's there. And then you put that into an electron microscope and look, and you can find it. All those are rather difficult things to use. If there's something that the virus produces in this culture, you don't necessarily have to see all the dead stuff. You just can take off some of the liquid it's growing in and test it. And one thing you can test for a retrovirus is reverse transcriptase. And it just happens to be that's a laboratory test available for it, so you just take a little bit off, put it into a chemical assay, and you can do it very, very simply. So it's a matter of something that you can put a lot of specimens through, and something that's simple to do, so you can really get maybe not a direct picture of the virus, you can't see it, but you can get evidence that it is there, like fingerprints.
Then he took the lymphocytes, which originated from Bruce's lymph nodes. He took these lymphocytes and cultured them with the lymphocytes of a healthy blood donor. And there, again, he detected reverse transcriptase activity. So Montagnier found reverse transcriptase activity, and according to him, this proved that the retrovirus was there. But the only way to say that the existence of reverse transcriptase or the detection of reverse transcriptase activity proves the retrovirus was there is only if reverse transcription was specific to retroviruses, which is not the case. In fact, today nearly everybody accepts that reverse transcription or reverse transcriptase activity is non-specific to retroviruses. In fact, at present, everybody accepts that reverse transcription is present in all normal cells. In fact, as far back as at the beginning of the 1970s, Gallo himself have shown that normal cells, when put in culture and they're stimulated or they're cultured with PHA, they will start having reverse transcriptase activity. Montagnier put the Bruce cells in a culture. Into culture, he added different growth factors, including PHA. And after 15 days, he detected reverse transcriptase activity. However, PHA by itself in normal cell, and this was known by Barré-Sinoussi, the principal author, and Gallo proved it at the beginning of the 1970s, that PHA itself causes reverse transcription in normal cells. So he put something in the cell which was causing reverse transcription, and yet he said that this proves the existence of a retrovirus there.
So you're saying that what they found might just be the actual substance they put in the culture and not a virus?
Definitely. That is their evidence. And
why would they do that?
Barré-Sinoussi knew that in 1973. Gallo proved it at the beginning. In 1972, I think, was his paper. I don't know. Now, everybody accepts that reverse transcriptase activity is a characteristic of retroviruses, but it's not specific to retroviruses.
But in all my interviews with scientists, they all say that reverse transcription is unique to retroviruses, and that's how they knew that there was a virus in their culture.
Once you have produced, you produce something in the extracellular medium, you can do actually several things. One thing is that if we expect or suspect it's a retrovirus, like HTLV1, the leukemia virus, what we can do is look for what we call reverse transcriptase activity, the enzyme, which is unique to these viruses.
Yeah, the scientific procedure was multifaceted. I had a good-sized lab, and so we had divided people up into different primary goals and would meet daily. And the first goal was, if a patient has fully developed AIDS, forget risk groups at the time. We didn't want to get into the risk group. They had AIDS, documented AIDS. Could we take their blood, get their T cells, like we would do with any, like the leukemias, like we did with the leukemias, the same protocols we did with leukemia. Take those T cells, clean them up from other cells, grow them with interleukin-2, our old friend, and see if we could find any evidence of release of virus particles, and if we were right that they would be retroviruses. And how to do that was with a surrogate marker. You don't use electron microscopy in those days, except maybe 1 or 2 pictures just to confirm or to see the structure of this particular retrovirus. The assay was reverse transcriptase.
Well, I don't know whom you interviewed, but if you don't believe me, go and ask Baltimore or Vermont, and I'm sure they will confirm that reverse transcriptase activity is a characteristic of retroviruses, but it's not specific to them.
What do you mean when you say reverse transcription is a characteristic of retroviruses, but not specific?
Oh, let me give you an example. Hair is a characteristic of humans. Black, white, or yellow, we all have hair, but it's not specific because there are many animals, for example, cats, dogs, which also have hair. And finding a hair in a room, it doesn't mean that a human being was there, or a cat or a dog.
Are retroviruses the only ones that can reverse transcribe?
No. There are other forms of reverse transcription that are used in various ways inside the cell. For instance, the ends of chromosomes are made by a reverse transcription process. That's how they're maintained stable. There is reverse transcription in the inheritance of all of our cells, which comes about from endogenous genetic elements in the cells, or in the cells of our ancestors, because once that information gets into our cells, into our genomes, it stays there forever. So it could be that we've inherited information from monkeys or from other animals that are in our lineage. And so, no, reverse transcription is actually very widespread. Something like 50% of the DNA in our cells comes about by reverse transcription.
Oh, that much?
Yes.
Oh, wow. Okay. That was 1 to 2%. Going back to, I thought it was 1.5 to 2%. The 50%, the retroviral DNA, or retroviral genes in our DNA, what biological function do they serve?
Now, we got to make a distinction. When I said 50%, I'm saying 50% of the DNA came about by reverse transcription. But it's not all retroviruses. Lots of it is just repeated elements that are there as what we generally consider to be parasitic DNA. DNA which is, other people call it selfish DNA. DNA which is in there because it's able to copy itself and reintegrate itself in other places. And this is something that's going on all the time. And it builds up.
Why do you think they used reverse transcription to prove the existence of a virus if they knew that it wasn't specific to viruses?
I don't know. It's as simple as this. I don't know. In fact, they went one step further. In the second experiment, they have taken the cells from their patient and mixed them with cells from a normal blood donor. And in that culture, again, they put PHA and other growth factors, substances which make the cell to survive and grow. And again, they found reverse transcriptase activity. And this time, they claim that the finding of reverse transcriptase activity in the second culture proves that the virus was transmitted from the patient's cells to the normal donor cells. And in fact, they say that this proves isolation of HIV.
But they must have used some other criteria to state that they had a new virus.
There was no other criteria in the first. They had a third experiment, but in the first and second experiment, that's how they are described in their paper. It was the first experiment, reverse transcriptase activity, the second experiment, reverse transcriptase activity. They are the only evidence they have published.
So in the
first and the second experiment.
So in both experiments, they fed the culture substances which artificially cause reverse transcription.
Yes. Yes, they fed them. You know, just one of them, PHA, by itself, it will cause reverse transcription in normal cells, in non-infected cells.
What are your problems with the third experiment?
Well, the problems, first of all, again, they took the supernatant, the fluid from their second experiment, and put it in a culture which contained umbilical cord lymphocytes. And in that culture, they also found reverse transcriptase activity. Again, the reverse transcriptase activity, they already use there this PHA and other substances which can cause reverse transcription in any culture, if the conditions are right. Then we have the virus particles. They have published electron micrographs showing virus-like particles budding from the cells and released in the supernatant. But just seeing virus-like particles is no proof that they are viruses, because you can have virus-like particles and they are not viruses. In fact, the third problem with the virus, with the particle, is that they use umbilical cord lymphocytes, which originate from placenta. In placenta, it was known in the 70s that they release virus-like particle and they have reverse transcriptase activity. So find them in this culture is no proof that they originated from Montagnier's patient.
In Montagnier's paper, they publish electron micrographs of HIV.
No.
We see pictures of the virus.
We see pictures. These particles were in, as I said, in umbilical cord lymphocytes. and umbilical cord lymphocytes originate from placenta. And there was already a lot of evidence by this time that placenta can release virus-like particles. What's more is Montagnier, when he described the particles he has seen, he said these particles are typical type C particles. Viruses, retroviruses are classified in different categories, one of which is type C particle. And they said that the virus they have seen is a typical type C particle. Typical type C particles have been many times published in the 1970s from normal placenta. In fact, most of the placentas was shown to have reverse transcriptase activity and typical type C particles. So finding these particles there, even if they are virus, they could have been just originating from the normal placenta. So finding the particles and finding reverse transcriptase activity in the umbilical cord lymphocyte culture does not prove that the patient was infected with a virus.
The two main things that told you had a retrovirus was the reverse transcriptase activity and the fact that the cells died.
And then we immediately call our guy who was responsible for electron microscopy and said, please, could you look on them under the microscope whether you can see virus particle and if it resembles to a retrovirus.
Okay.
Okay.
And what did you see? What did you guys see when you looked under the electron microscope?
And after quite, it was very difficult because it was only few cells infected, so it was a very difficult task for him to find a cells that was just producing this particle, but finally he found it. And he found one lymphocyte with a budding particle, typical of retrovirus, and very close from these cells, one complete mature particle that resembled to a retrovirus.
In that paper, he had only one electron micrograph, and the virus could be identified as sort of a retrovirus, but it could have also been an arenavirus. Now these are specifics that are only important for experts. But when we saw that photo, we said suggestive, but not convincing.
So I've seen these publications, stamp-sized images. It's a nuisance. It's a nuisance. You do not really see much. And I do not know how good electron microscopy was really done, but they probably should have done a bit more, and then they would have been very safe. And electron microscopy is an art and in science. Art and science, and nobody is able to judge what the electron microscopist is really doing. You have to have experts discussing these things. Sometimes people are far from the microscope. They do not understand structure and function. They like to have a nice image, that's all. They do not understand sometimes, and that's a danger. I could imagine if Montagnier would have pushed the structure, the fine structure of HIV a bit more, he would have been more convincing.
Would I be right in saying that from the pictures alone, though, in culture, you can't prove what virus you found yet?
Of course, electron microscopy is not sufficient to prove you have a retrovirus, that's clear. You need other characteristics like the density. The reverse transcriptase activity is the key enzyme, which is at that time was only associated with retroviruses. You know, that's how we describe it first. You know, we didn't describe the electron microscopy as a proof. We described the reverse transcriptase activity as a proof it was a retrovirus. But here also we were criticized by some people from Gallo's lab saying, are you sure you have a reverse transcriptase activity there? Could it be a cellular DNA polymerase? You know, we had this kind of question.
So Montagnier, he wanted to show not only that he had a retrovirus, but that the retrovirus was new, was not one of the two other retroviruses which Gallo claimed to have discovered at the end of the 1970s and the beginning of 1980s in leukemic patients. For this, first of all, he said he purified HIV. He obtained, by purification, we all agree, he said, and Montagnier himself admits that to prove that a virus is a new virus, then you have to show that it has proteins which were not present in any other retrovirus. And to do that, he said, you must purify the virus. He admitted that in 1997, that you must purify the virus. And in 1983, he claimed to have done exactly that, to have purified the HIV particle. Now, we may have differences as to what isolation means, but when it comes to purification, there is no disagreement. By purification, they mean to obtain particles, retrovirus particles, separate from anything else, which at least from anything else which contains proteins and RNA.
Now, when this virus is in this supernatant, it's not purified. Okay, because the cells are releasing plenty of things, not only the virus, cellular protein and so on, okay? Yes. So that means that in the supernatant you have a mixture of everything, including the virus. Then you have to purify.
Okay.
Okay. This is the second step. Then you try to purify the virus from all this mess. When we started in the early 80s, we were trying to purify the virus by a sucrose gradient.
Retrovirus particles are purified using a laboratory procedure developed over 40 years ago known as density gradient centrifugation. The cell culture that is undertaken by the scientist to produce workable quantities of virus results in a liquid suspension made up of cells, macroscopic and microscopic cellular debris, virus particles, if any are present, and culture fluids. This suspension is spun in a low-speed centrifuge which creates a sediment consisting of the cells and heavier solid material, and, above the sediment, a liquid supernatant containing the much lighter microscopic material. If retroviral particles are present, this is where they will be distributed. Next, a small portion of the supernatant is removed and very gently placed on top of a solution of sucrose. This sucrose solution is prepared in a special way such that its density increases gradually from the top to the bottom of the tube. In this diagram, the layers of different densities are shown as discrete bands, but in the real world of the laboratory, these layers gradually merge into one another. Purification by this technique relies on the fact that particulate matter in the supernatant sample will gradually sink down through the sucrose solution until it reaches a place in the gradient where the sucrose solution and the particulate matter have the same density. When an object gets to this portion of the gradient, it cannot go any further. It is exactly like trying to force a tennis ball to stay put at the bottom of a bucket of water. As soon as you let it go, it bounces back up to where it wants to float according to its density in water. This means that in the sucrose density gradient, all objects of the same density will eventually congregate at the same place in the gradient. In the case of retrovirus particles, this is where the density of the sucrose reaches 1.16 grams per mil. Because the particulate matter in the culture supernatant is so light and tiny, the passage of the sample through the gradient has to be speeded up by spinning the tube in another kind of centrifuge known as an ultracentrifuge. This machine rotates the tube at speeds between 40 to 60,000 revolutions per minute and produces a force many thousands of times gravity. In this diagram, we have assumed the supernatant sample contains retrovirus particles, and you can see them gradually moving through the gradient and being arrested at the 1.16 grams per ml density. Here is a short demonstration to illustrate how a sucrose density gradient solution can be used to separate objects of different densities, in this case beans and macadamia nuts. In this simplified version, we will use a two-band density gradient consisting of water with a density of 1 gram per ml and sucrose at a density of approximately 1.5 grams per ml. First, we prepare our sucrose solution by dissolving sucrose, ordinary table sugar, in water. Then we make our two-density layer solution by carefully pouring our lower density layer, the water, on top of the denser sucrose layer. Next, we place our sample on the top of the two-layer solution. Since these objects are so much larger and heavier than virus particles, gravity is more than sufficient to propel them through the gradient. In fact, as you can see, in this experiment the separation is virtually instant. There is no need to spin the glass in a centrifuge. The result is one density band consisting of nothing but beans, and another density band consisting of nothing but macadamias. In other words, these objects have been successfully purified. What may not be obvious is the part your eyes play in this experiment. Without looking at the two density bands, which is equivalent to performing the electron microscopy, you will not be able to tell if any objects are present, what is their morphology, and whether or not they are pure.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus,
So Montagnier claimed to prove that he had particles, he had proteins which are not present in any other retrovirus. He purified the virus. But he did not publish any electron micrographs of the purified material to prove, you seeing is believing. And they claimed that they had purified material, but there were no electron micrographs. And this is one of the conditions where Barré-Sinoussi and Jean-Claude Charmaine put it in 1973. You must have picture of the purified virus and show that the material contains nothing else but particles with the same physical characteristics. But no such pictures were published.
Is it important to photograph where the virus is banned in the gradient?
Yeah, because retrovirology has also some history. These are established techniques. If you go to C-type particles, they can be easily bended and they are stable. And if you think HIV is a new retrovirus, you have to go the same way, just to be acknowledged as a retrovirologist dealing with that new virus in a proper way.
Hans Gelderblum, he said it's important to photograph the density gradient where virus is banned. Why is that an important step?
Because this is, electron microscopy, for example, it's always very important to see the morphology of the virus particle. It's in the region of the density gradient, okay, where you think you have a virus purified, and I will say in good shape. If you check after by electron microscopy, the electron microscopy will tell you, okay, you have the right structure of the virus particle with all the envelope which is on the surface of the membrane, okay. Because very often when you make purification, you alter all this process, making sucrose gradients and centrifugation. It's forces, on the virus, pressure on the viruses, and it does not like it too much, especially for HIV, where the envelope protein, which are anchored on the membrane, are not covalently anchored. So that means that very easily you can lose the envelope protein.
Oh, the GP120?
Yeah. So it's important to check then on the electron microscope whether the structure of your virus is the one that you are looking for, with all its viral protein, the envelope protein, structural protein, everything. You have the right shape, the right structure.
And it also shows you what isn't there, correct?
Exactly, also.
So you're saying Montagnier never proved the existence of a new retrovirus because he didn't photograph in the test tube?
Or, as I said, he says it is essential to purify the virus to prove that there are, it has proteins which are not present in any other virus. That's the only way to prove that you have a new virus. But he did not publish pictures. So since he did not publish pictures, we don't know what he had in his purified virus. It is possible that he had purified virus, but it's possible that he did not have anything there. And that's what we've been asking from the very beginning. Why there were no pictures, which are essential to prove purification.
When purifying, Gelderblum told me it's important to photograph the density gradient where virus is banned. Why is that a crucial step?
Well, Gelderblum, I know him well. He's a good electron microscopist in Berlin. And actually, he gave me the best picture of my virus.
Oh, really?
Yes. Well, of course, in order to purify, you have to make this sucrose gradient density equilibrium to have a sharp band. And if you take that sharp band, you have almost pure. Not completely pure, because there are cell vesicles which have the same density. This is why you don't see in the picture you have a mixture of cellular vesicles, and viral particles.
So, is it really important, or is it not really important?
For us, it's not important. But some people say if you don't have complete purification, how do you know the disease is not caused by something else?
To silence them, how come you guys didn't just show pictures from the gradient instead of just the culture?
We first show it from the culture. Just by centrifugation, but not sucrose gradient, just by making a pellet of the virus, you could look at it also this way. But here, of course, you have many impurities, coming from the cell. The sucrose gradient has the advantage to partly purify the virus, but again, even in the band of the virus, you have also cellular vesicles, which have the same density, but not the same look, of course, at the electron microscope.
And you're saying that in the purified banding area, there can be other contaminants, and that's why pictures are essential?
Yes, in the purified, the method they used, you can get, by this method, you can get material which is not viral, but it has proteins, it's cellular fragments. The cellular fragments can, with this method he's using, could be at the same place. And then you can have there only cellular fragments, or you can have a mixture of cellular fragments and viruses. But it's important, it's crucial to have a picture.
If you separate by density 1.15 gram per ml, you have a lot of vesicular stuff inside, not related to virus. Detritus, a reaction product of the cell. You have a lot of host cell constituents in that band. That's not too nice.
So are there other particles that can look like retroviruses in that band?
Yes. In that material you can have cellular fragments, and they have proteins and they have RNA, which retroviruses have, and in fact they may even look like retrovirus particles. So it is important to have, it is crucial to have an electron micrograph of the material for us, for example, and from any other scientist to believe what they are claiming.
When you purify HIV, there are some challenges because it's contaminated with cellular debris.
As I said.
And particles that look like retroviruses but are not infected.
Yes, as I said.
How do you distinguish between what is infective and what isn't?
You cannot.
Montagnier gives crucial importance to this band, because he said if the particles do not bend at the 1.16 gram per mil band, then they are not retrovirus particles.
The world accepted those papers and the pictures that he did present as being evidence of a new virus.
Well, the world accepted these pictures, and the world accepted that Montagnier proved the existence of HIV, but this is what we have not accepted from the very beginning. We've been questioning, we've been asking in scientific papers, and today neither Montagnier or anybody else has responded to these questions.
Isn't there a chance that you're wrong, since everybody concedes that Montagnier did isolate HIV and purify it?
Well, who's seen it? We're giving you the evidence. And to me, this evidence, and to my colleagues, this evidence does not prove the existence of HIV. Now, most of the people may have not even read the Montagnier paper, and yet they accept that Montagnier proved the existence of HIV. We mustn't forget that doctors are very busy, and it is very hard for everyone to go and study in detail all the claims. You have to really put a lot of effort to put all the things together.
But it's been 26 years since Montagnier published his papers. In 26 years, billions of dollars have been spent on HIV research. So doesn't the fact that, isn't Montagnier's paper kind of antiquated? I mean, hasn't it been proven that HIV does exist in the past 25, 26 years?
Well, nobody has presented any better evidence for the existence of HIV than Montagnier and Gallo. Gallo presented similar evidence. Apart, the difference between Gallo and Montagnier was that Gallo used, instead of umbilical lymphocyte T cells, he used the leukemic cell line. But otherwise, they published the same findings. And their findings today, still even today, are the best evidence anyone has presented for the existence of HIV.
So let's go to the test tube. What did Montagnier find in the purified band?
Montagnier took the proteins from that band, which he said, or the material he said was purified HIV, and then he reacted them with sera from his patient. And in the purified material, he found 3 proteins which reacted with antibodies which were present in the sera of his patient. P80, P45, 41 now, and P24. He said 41 was a cellular protein which contaminated his virus, made no mention of P80, and he said P24 was viral. Well, the question is, why only P24 viral and not P80? From antibody-antigen reaction, you cannot prove the origin either of the antibody or the protein. And he did not have evidence that P24 was coming from a particle. In fact, we did not know that there were any particles there. And then he said, he defined it by this reaction, that P24 was viral and the antibodies were viral. This cannot be done scientifically.
HIV is said to have 9 proteins.
Yes.
But Montagnier only found 1 protein
in
his purified culture.
That's one of the big questions. If what he had there, what he called purified virus, it was HIV, and so it will have had all the proteins which a virus has, which HIV has, 9 or 10 proteins. And the patient, the antibodies which reacted with the P24 should have been also antibody to all these other proteins. And yet, the patient didn't have any antibodies to react to the other proteins, and we don't know if he had any of the other proteins. So this is a big question. You cannot have a virus which has only one protein. The question is now, if that was a protein, if he had purified virus, where were the other proteins? And if the patient was infected with the virus, where are the other antibodies? How can we have only one protein and one antibody? It is not possible. But unlike Montagnier, who considered P24 the only HIV protein, and 41 as being a cellular protein, Gallo in 1984 considered 41 as the most specific HIV protein and not a cellular protein.
So maybe Montagnier was just wrong and Gallo was right.
Unless you know that you have a purified virus, unless you have evidence that there was a purified virus, you don't know which one is right. Because Gallo did not have any electron micrographs. Gallo could have said that 41 is HIV if and only if he had published electron micrographs and showed that what he called purified virus had nothing else but virus-like particles. Now, the next criticism is that Montagnier's particles, he said that his particles are typical type C viruses. Then, in 1984, he said that his particles were type D particles. In Levy, at the same time, said that they were type D particles. Gallo, again in 1984, said that the particles he's seen were belonging to the family to which his previous viruses existed. And so, and they are type C particles. The two retroviruses Gallo claimed to have discovered, they were type C particles. And then, from about 1985, 86, most of the HIV experts start saying that HIV is a lentivirus belonging to a totally different subfamily of retroviruses. And even today, there is still disagreement as to what subfamily and species the HIV particles belong.
Anywhere you travel in the world, you're going to find somebody that generally looks like somebody else. And when I look at electron micrographs, all viruses look the same to me. So is there really a difference between whether it's a C or a D or a lentivirus? Is it that big a deal?
Well, there is a big deal because they belong to different, according to retrovirologists, they belong to different, not only to different species, but to different subfamilies. And this is not different than saying that people saying that they see, one and the same thing, and yet what they see, some see a human being, others see a chimpanzee, and others see a gorilla. The difference is that much. We all belong to the same family. Chimpanzee, humans, and gorillas belong to the same family. All the retroviruses belong to the retroviral family. But we are in different species, and so are the HIV particles.
Would a trained eye be able to look under the microscope and easily distinguish between a type C, a type D, and a lentivirus?
That's what they say. That's what the electron microscopy say.
Using the electron microscope, how easy is it for you to differentiate between all these retroviruses? Because they all look, to someone like me, the untrained eye, they all look the same.
Certainly not. I will be able to teach you within half an hour. We have made a nice summary on these structural differences. You can measure. And if you start measuring the ribonucleoprotein, that's a complex of the genome and some protein, where is it in the virion, in the budding, in the immature particle? How is that organized in the mature particle versus the immature particle? What about the glycoproteinops? These are questions that can be quantitatively assessed. So you really can make an objective diagnosis.
So for someone like you, it's easy to tell the difference.
Yes.
The cone-shaped core is very identifiable. It looks very different than a C-type, is that right?
Yeah, absolutely.
Okay, or at least to the trained eye.
No, you will see it.
Oh, okay. How come, in your opinion, did Gallo and Montagnier see C-type instead of lenti when they looked under their electron microscopes?
Because they had it in the brain.
Oh, they saw what they wanted to see?
Yeah.
Oh.
Still, the purification is the biggest problem, the purification. Once, if you don't have purified, if you have no good purification, as Montagnier said, if you have no good, you got to have purification to be able to characterize the virus. Even if one assumes that Montagnier and Gallo and the particles are retroviral particles, the reverse transcriptase is specific to retroviruses, to say that you have a unique retrovirus, you must characterize, you must its proteins and its RNA. You must show that these have proteins and RNA which is not present in other retroviruses.
And why, in your mind, is there such inconsistencies in identifying the virus among the experts?
The particles?
Yes.
I don't know. We cannot say that you have a new retrovirus unless you show that it has unique proteins and unique RNA. And to show that, you must purify the virus. There is no other way. If you say that these proteins and this RNA are HIV RNA and HIV protein, you must somehow obtain them from the virus particles. But because the viral particles are so small, the next best thing is to obtain them from a mass of material which contain nothing else but retrovirus particles.
Well, when I talked to Flossie Wongstall, she said that you don't necessarily have to take pictures. You can go to the culture and look for viruses budding as evidence of release of virus.
Yes, as I said to you, budding and retrovirus-like particles, just seeing them in the culture, there is no proof that they are retrovirus. That is no evidence. Take pictures from the culture is no proof that they are virus. And certainly you cannot prove just looking at that. There are so many things there in the culture which also contain proteins, which also contain RNA and contain DNA. To say that HIV has 9 proteins and HIV has a genome, a unique genome, 9 genes, you must have evidence that these proteins originated from the viral particles. And to do that, you must take these particles out from the culture. You got to have them out. You must purify them. You must obtain them separate from everything else which contains proteins.
She said that there were problems that when you spin the particles under the high-speed centrifuge,
yes,
that they often distort, or they lose their envelope, or they break apart. And that's why it's almost impossible to purify viruses.
No, there are many pictures of electron micrographs of retroviruses which have been purified. You know, I can show you, including this one, a sarcoma virus. When you take somebody to court for a paternity suit, you must have evidence that the blood originated from the father and from the child. There is no other way. You must have proof that they originated from the father and from the child to compare the DNA. The same way, if you want to say that these proteins are from HIV, you must have them coming from HIV. But the particles are too small, so the best next thing is to purify viruses. And this is not me who says it. I mean, this is all the virologists, including Montagnier, including Charmaine, including Barré-Sinoussi. That is, this is so simple. So you must have purification. That is the only way. You see, this is what the HIV. Retroviruses have the matter of 100 to 120 nanometers. They have a cone-shaped core. They have the lateral bodies, and they have knobs.
What are the knobs?
When we see HIV being diagrammatically represented, you see there's some spikes coming out from the particle. And these knobs are extremely important for effectivity. Again, according to all the HIV experts, if you the knobs are crucial. They are essential for infection. No knobs, no infection. And there is no infection, the particles cannot be viruses.
So a complex process of an interaction of the outer protein of the virus, called the envelope, with molecules on the surface of the cell are essential for a virus to enter the cell. And when we speak of entry, we essentially mean the guts or the internal component of the virus that contains its genetic information in the form of RNA and certain other proteins.
Today, nobody has proven the existence of knobs in the cell-free particles.
Obviously, when we first were working on the cause of AIDS, we had to be very broadly, have our ability to include a whole variety of different potential organisms that could cause it. What was helpful in the limiting of our search were the cases in hemophiliacs where the material that's used to treat hemophilia comes from blood or plasma, a subcomponent of blood that is sold or donated into the system, and then a purification process is used to take that large volume of plasma and specifically pull out the anti-hemophiliac factors that actually can treat the hemophiliacs. Now, in that process, the one thing you have to realize is that material is given intravenously to the hemophiliacs to treat them, and you have to have it very clean. You don't want to have any infectious agents in that if you can avoid it. And one of the easiest ways to do any of preparation of a drug is to filter it. Now we have these wonderful modern filters where you can work on your drug, and then the last step before you put it in the bottle, pass it through a filter that filters out all infectious agents. Now I say all, meaning that the filter can only filter out those that the filter can catch, which is the bigger agents. That bacteria and parasites are much bigger than viruses. And those filters indeed will filter out all the bacteria and make it, quote, sterile. That is, you cannot grow bacteria from it. But yet we knew that after this process that they were still getting this disease, which could have been caused by a virus, a bacteria, or parasitic disease, or whatever. But since all of the larger bacteria and parasites are eliminated in these filters, then you can assume that the disease that you're looking at is caused by a virus.
And does it also filter out the infected cells? So is it just pure free-floating HIV virus?
The factor VIII material is cell-free, so it's only liquid material, and that liquid material could contain many infectious agents from the donors of all these. Literally, there's hundreds of donors in each of these batches. And when you filter it out, you filter out all the bigger organisms, that is, bacteria and parasites. You're only left with viruses that go through the filter.
I reckoned that a virus that would survive the purification of clotting factors from human blood was more likely to be a virus without a cell membrane, a small virus like a parvovirus, and I would not have put a retrovirus at the top of the list. And yet, when HIV was discovered, it was indeed a retrovirus. I think my scientific reasoning was perfectly correct, but I was deluding myself nonetheless.
Is there something harsh about the factor process?
Well, I didn't realize how unharsh the purification of clotting factors is. We were talking about purification. Those clotting factors are anything but pure, and they were tainted with this virus. They were tainted with hepatitis B virus, with hepatitis C virus, which also was not discovered until long after HIV was discovered. So HIV was just one of several viruses that was passed in clotting factors.
Here it is a paper which was published by several authors, with a co-author Geldenblum, and they say they have found that on the average, immediately after release, after the knobs are released, the particles into the fluid, there are 0.5 knobs per particle. And they said even these 0.5 knobs, they may be false positive. That is, there are no knobs at all.
The knobs here look like little knobs everywhere.
Yeah, okay. There's a chance that you have some dirt, of course, in the cell culture. And these would be knobs from a particle that is cut tangentially. Here the same. That's my interpretation. So my interpretation. But here are the knobs. Here might be there might be some dirt. Here, sure, that is dirt from preparation.
Mm-hmm. But don't these look like knobs too, coming off of this big one?
Yeah, you know, there are glycoproteins also on the cell.
Okay.
Yeah. Now, we have to differentiate between them. If we would like to go for differentiation, we have to use immunoelectron microscopy to make a tag. It's either the glycoprotein or it's something different.
In a paper published in 2003, the author stated, the cloth of GP180, that is the knobs, do not form spikes on the surface of HIV, as is commonly described in the literature. We suggest the spike knob observed by negative staining electron microscopy may be an artifact of the penetration of heavy metal stains between the envelope proteins.
What exactly does that mean?
That means that despite some people claims that there are knobs on the HIV particles, these authors say that the particles have no knobs. In plain language. This is a paper published 2006 in Nature. Here it is SIV, Semen Immunodeficiency Virus. And in these particles you can see many knobs. But when you come to the electron micrograph of what is meant to be HIV particles, there are hardly any knobs. In fact, we can see only one there, but you can see the same thing down there. And you don't know if there are knobs or there is some artifact there. In fact, the authors call them putative knobs. Or putative means supposedly HIV knobs. Mm-hmm. So the authors do not have, even today, proof that the HIV particles have knobs which are crucial for infection. Even if we admit, even we accept that there are knobs, there are 0.5 knobs immediately per particle on the average, immediately after they are released, they are lost. Even this small number is lost very rapidly, according to Geldenblum. And these knobs being lost so rapidly, and factor VIII preparation taking a long time, from the time the blood is taken to the time factor VIII is prepared, is a long time. and even after it is prepared, may stay on the bench for months before it is used. So it is impossible for hemophiliacs. The factor 8, to contain an infectious HIV particle, even if HIV exists.
I wanted to ask you about hemophiliacs. Because they had cell-free plasma, it was just the virus. But the virus sheds its membranes within 24 hours. So one thing we couldn't get is how was it able to actually infect the T cells?
Yes, it's a question, but we have to know that all the fraction of the blood could be infectious. And there is some virus bound to red blood cells which could be released also in the plasma after treatment or incubations. Some virus is bound to red cells also as well. So perhaps there are more virus when you process the blood. More virus could come in the plasma. Okay, and these virus could be protected by the plasma proteins from denaturation. This is one thing possible, one possibility. The other is the quality of the host. Hemophiliacs are fragile. Being transfused many times, their immune system also is depressed before they get infected with HIV. So they are prone to HIV infection because of their immune system weakness. Okay. So combining this with the fact that the virus could exist also in forms which are studying now, which probably are more resistant than we think for the usual particles, could explain.
The little ones.
Yes, little ones, or they bound to macroplasma proteins. You know, that's possible that macroplasma are more resistant, perhaps. Macroplasma envelope could be more resistant than the viral envelope.
Okay.
But there are all hypotheses. This is not based on solid data, of course. This is just they are just assumptions. But you are right. We have to explain why hemophiliacs have been so easily infected with plasma products.
For you, the entire existence of HIV rests upon the fact that there are no pictures of the purified gradient. Is that correct?
It is part of it. That is the most crucial evidence which you need. If you don't have these pictures which prove that there are, in the purified virus, what they call purified virus, they are virus-like particles, then the whole experiment and thus the existence of HIV is finished.
And you're saying to date there is no pictures of purified virus?
Today there is no pictures of purified virus. And certainly, Montagnier did not publish it, Gallo did not publish it, Levy did not publish such pictures, Weiss did not publish such pictures. And the only pictures which have been published was, in fact, this is admitted by the Franco-German researchers in 1997, when the first attempts, the first pictures of what is called purified HIV were published by two groups, one from the United States and one in a Franco-German study.
You said in 1997 they did try to purify HIV, is that correct?
Yes.
And you're saying they weren't successful?
They are not successful. And what is more important, these authors, they accepted or they admitted, but by 1997 there is no evidence for purification. And here it is. This is from the Franco-German study. Virus to be used for biochemical and serological analysis or as an immunogen, that is as an antigen, is frequently prepared by centrifugation through sucrose gradients. And they said, in none of these studies has the purity of the virus preparation been verified. So by 1997, there is no proof that HIV has been purified.
But did they purify HIV?
They tried. So they accepted this as I said, we've been asking this for the very beginning to have some evidence for HIV purification. And these authors tried to present, or they did their best to purify HIV. And here is their evidence. This is the Franco-German study. And they had material which is meant to represent purified HIV. The top and the middle is material obtained from infected cultures, and is meant to represent purified HIV. The bottom part is material obtained in a similar manner from non-infected cultures. And as you can see, the arrows, they said, represent the HIV particles. First of all, this cannot be said to be purified virus. As you can see, they are not purified particles. In fact, only the particles which they put the arrows are said to be particles which look like HIV. The rest are all cellular fragments, or called vesicles. In the material which is obtained from the non-infected cultures, you can see even there are some particles which may look like the one which is arrowed. So it is significant that the authors do not call this material purified HIV. In fact, they call it purified vesicles from infected H9 cells, the top, and activated cells. So the authors admit that this cannot be considered purified HIV. With all the effort they put, they could not obtain purified HIV. Okay. It is also important that the particles which are arrowed as representing HIV, they don't have all the morphological characteristics attributed to HIV. There is no evidence for knobs. In fact, even their diameter is higher than what is considered to be the retroviral particles.
But does size really matter? I mean, humans vary in sizes.
They matter because this in the Franco-German is the size is, they are larger, but not as large as in the American evidence for purification. Here is the effort by the American researchers in purifying HIV. These are their results. And as you can see, they were not able to obtain purified HIV. And again, the particles which are arrowed and are said to represent HIV, they do not have all the morphological characteristics attributed to lentiviruses. And in fact, they don't have the knobs, they don't have cone-shaped core, you cannot see there, they don't have lateral bodies, which are, they should be in HIV, and most importantly, their diameter is they're too large. The average diameter of the particle is 234, and none has a diameter less than 160. Just by taking their diameter, it is impossible the particles which are labeled as HIV to be HIV.
Is the size also important in determining what you're looking at?
Yeah. Think about viruses can be as big as pox viruses, up to 300 nanometer. HIV up to 150. The smallest autonomously growing virus will be a circovirus, just 15 nanometer.
So what's the smallest HIV particle that's ever been documented?
I think it's something 120 to 150. No, these are fixed morphological entities.
They don't change.
They don't change.
So that's a huge thing in helping you differentiate whether it's HIV or not.
Yeah. The size of a structure is very important to make the diagnosis.
Now, if the particles which are labeled by Bess and his colleagues as being HIV, then the absolutely necessary condition is for the material which was obtained from the infected cultures to have proteins which are not present in the material which was obtained from non-infected cultures. Right. But this does not seem to be the case, and in fact, Bess and his colleagues have come with this evidence. They took the proteins from all the three bands they had, from the two infected cultures, or what they called infected cultures, and from the non-infected cultures. And here are the proteins from the non-infected cultures, which they are put in a strip. Here are the proteins from the two infected cultures. As you can see, if you look at this, there is a difference, but the difference between the strips is only quantitative. That is, we have these bands in all the strips, only there is less here. Similarly with all the other proteins. In some of them, in fact, they are exactly the same. So, by looking at these pictures, the proteins which existed in the purified material, the HIV purified material, and in the material which was obtained from the non-infected cultures contain the same proteins, which means that none of these cultures contained HIV. As I said, if the cultures which contain HIV, they must have had proteins which are not present in the non-infected cultures. And yet, this is not the case. The proteins, all the proteins are found, which are found in the so-called infected cultures, they are also found in the non-infected cultures. Some of them in smaller quantities, but nonetheless, they are there. The difference may be just because the way the cultures were conducted. The American authors labeled some of the proteins, and the P6, P7, P17, and P24 are labeled as HIV proteins. And these two proteins, that is, proteins which are around 32 and around 41, they were labeled as cellular proteins. No label is put in the proteins which had molecular weight higher than 41. Yet there are many HIV proteins which have molecular weight. Now, why they didn't label them? First, all the proteins which are around 41 and 32 are non-HIV. There cannot be HIV proteins with molecular weight 41 or around 41, a molecular weight of 32 or around 32. Okay. We asked Bess why they labeled these other three proteins as HIV. And he said that they put this label because that's what the reviewer asked them. But they did not have evidence themselves that they were HIV proteins.
He stated that they did not have evidence that they were HIV proteins?
Yes, he stated that they did not obtain themselves, but the reviewer asked them to label them as HIV proteins. And he said the reviewer was right. We labeled them HIV.
Did he specifically state they had no evidence those were HIV proteins?
Personally, they did not have evidence. Yes.
Bess said that?
Yes. In this experiment, they did not obtain evidence. In this experiment, they did not obtain evidence that these proteins were HIV. But they labeled them HIV because the reviewer asked them. So the question is why they didn't label the proteins higher than 41. Well, there are some good reasons for it. First of all, many HIV experts, maybe not all of them, but many HIV experts accept that the protein of 120 and 160 are polymers of P41. That is, they are 41 joined together. 3 41 are joined together and are made 120. 4 41 are joined together and make you 160. They admit all the proteins above 24, there is evidence that the protein above 24,000 molecular weight are cellular proteins. And they also know that the proteins, well, is molecular weight lower than the P70 and P6, P7, they are subunits of proteins with a high molecular weight. So now we are left with one protein, P24. We come back. Now, what is 24? Well, to answer that question, we have to go to an interview which Montagnier gave to the French investigative journalist Jamel Tahi. Jamel Tahi asked Montagnier why they didn't publish any electron micrographs of the material which they said represented purified virus. And Montagnier responded, we found some particles, but they did not have the morphology typical of retroviruses. Jamel Tahi insisted to find how it was possible that they did not publish when they claimed that they have purified it, and then in the purified virus they did not have typical, even retrovirus-like particles, much less HIV and much less purified. Montagnier repeated, I repeat, we did not purify it. Then he was asked if Gallo purified it, and Montagnier responded, I do not know if Gallo, really purified. I don't believe so. Jamel Tahi asked Montagnier, do any pictures from the for purification exist? Montagnier responded, yes, of course. Have they been published? Montagnier, I could not tell you. We have some somewhere, but it's not of interest, not of any interest. For us, it's of very great interest. It's crucial. If you don't have them, then there's no way you can prove the existence of HIV. And then we asked Gallo. Montagnier subsequently published many pictures of purified HIV particles, as of course we did in our first paper. You have no need to worry. The evidence is obvious and overwhelming. Or there was no pictures of purified virus in Gallos's 1984 papers, or of any other of Gallos's publications.
Going back to 1983, when trying to prove the existence of a new virus, why was purification important?
It was important to prepare kits for antibody detection. Okay, because we wanted these diagnosis kits to be as specific as possible. Okay. If you use a preparation of virus which is not purified, of course you will detect antibody to everything, not only against the virus, but also against all the proteins that are produced in the supernatant. Okay.
Now, all these pictures here,
yeah,
are these all from culture?
All from culture.
Do you have any from the gradient?
Yeah, there's 80% of dirt.
Oh, 80% of dirt.
Yeah.
Oh.
And therefore I didn't like that, but it was necessary also for us to control, because this house in 85 already established ELISA antigen material for testing people. There was nothing commercially available, but we had to purify. We had to look at the material that was used for the ELISA. 80% dirt. Okay.
Okay.
That's the truth.
Again, Jabel Tahi interviewed Charles Dauger, which was the electron microscopist at the Pasteur Institute. And in his response, he said, in the purified virus, they had only cellular debris. They never had virus. Which means that the P24 protein originated from a material which contained not even retrovirus particles, much less purified virus.
So in 2001, the EM specialist from Pasteur admitted they never saw anything?
Yes. Yes, he admitted that they never saw any virus-like particles, much less purified virus. In fact, what they had there was cellular debris all along.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus, you know.
The P24 originated from a material which did not even have virus-like particles, much less purified virus.
So it's cellular debris?
Yes, had only cellular debris, which means you cannot have a better evidence that P24 is a cellular protein, cannot be HIV. If it originated from a material that contains only cellular fragments, it has to be a cellular protein. Now, even if we assume that P24 is an HIV protein and that Montagnier discovered the first one-protein virus, this creates even a bigger problem. This is because retroviruses have an enzyme called reverse transcriptase, which is a protein. That's why they are called retroviruses. Their name derives from this protein. If they don't have this protein, if they don't have this enzyme, the virus cannot be a retrovirus. That would be like having an object which does not have any wings, and yet you still call it an aeroplane. Now, we know that in his experiments, Montagnier found reverse transcriptase activity, and on the basis of this activity, he claimed to have proven infection of brew and his cell cultures with HIV, as well as detection, transmission, and isolation of HIV. Since P24 is not a reverse transcriptase protein, as you can see from the picture, it means that all Montagnier did was prove what they all knew all along. Reverse transcriptase activity is not specific to retroviruses. Under the right conditions, it can be detected in all cells infected with a retrovirus or non-infected with a retrovirus. If there are no HIV proteins, there can be no HIV genome. What Montagnier and Gallo called the HIV genome is nothing more than a form of RNA known as adenine-rich RNA, which they found at the 1.16 gram per mil band among other RNAs and DNAs, despite the fact that Gallo never published proof that the band contained retrovirus particle, and Montagnier admitted it did not have any, they called this RNA HIV RNA. It's even worse. Gallo knew as far back as 1972 that adenine-rich RNA is not specific to retroviruses. This type of RNA can be found in any cells which are synthesizing proteins. In any case, the existence of HIV and even its causative role in AIDS were both accepted well before any genomic studies were published. Today, we have not yet proof for the existence of any of the HIV proteins. And if you don't have proof for the existence of viral proteins, you cannot have proof for the existence of the virus.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus, you know.
How come you guys didn't just show pictures from the gradient?
We first show it from the culture. Just by centrifugation, you know, but not sequence gradient.
From the pictures alone, though, in culture, you can't prove what virus you found yet.
Of course, electron microscopy is not sufficient to prove you have a retrovirus, that's clear. You need other characteristics like the density. The reverse transcriptase activity is the key. We didn't describe the electron microscopy as a proof. We described the reverse transcriptase activity as a proof it was a retrovirus.
No, reverse transcription is actually very widespread. Something like 50% of the DNA in our cells comes about by reverse transcription. When I said 50%, I'm saying 50% of the DNA came about by reverse transcription, but it's not all retroviruses.
The problem is that they detected non-specific phenomena. Reverse transcription, particles, and we've seen all the problems with particles, and antigen-antibody reactions. And so you can't take a whole lot of things that might be something and turn them into that something. That's the problem. If you're walking down if you walk into an empty space and you find an engine block and a fan belt and a generator lying on the ground, what do you say you've got? Do you know what you've got? I mean, is that a car? Could it be a boat? Could it be a plane? Could it be something someone uses on a farm to lift grain up? It could be all those things. You can't make something specific out of a whole lot of things that are non-specific. So they used a whole lot of non-specific and just preferred to believe that this is what they'd found.
What Montagnier did is similar to a fisherman throwing his net into the ocean, pulling it back, looks at it, finds no fish, and yet he claims that not only that he has fish, but he has nothing else but fish, and all of the same kind, HIV.
nothing like HIV. There's no disease, no infectious disease that essentially kills everyone who gets it.
The fear factor's gone, and yet getting HIV infection is a problem for any individual. The fear should still remain because it's a virus that kills people.
There is this sense of complacency that it's better, and it isn't better. And all these people, these 40+ million people living with the virus, they will die of AIDS.
Over the past 3 decades, humanity has rallied together for the AIDS cause. People from all walks of life have united across all social and economic boundaries, joining hand in hand for one common purpose: to end AIDS.
And I want you to say the name of the person you're walking for. I'm walking for Robert Johnson. I'm walking for Rock Hudson. I'm walking for everybody that isn't here to walk for themselves.
The statistics are growing, all except for one. That's the amount of people cured, and it's still 0.
In theory, we could cure AIDS on a piece of paper. Maybe someday it'll come. I can't say when.
Despite all the major progress we have in chemotherapy of those patients, none of these patients got rid of the virus.
I don't think the pharmaceutical industry is very interested or invested in a cure. You know, I don't mean to be too cynical, but the reality, I think, is that, you know, the situation they've got now, which is lifetime treatment with expensive drugs, that kind of suits them pretty well.
A cure is going to require some very tricky and sophisticated molecular biology, and I frankly don't see it happening.
Ever?
Ever.
I was born in 1980, a year before AIDS exploded onto the public consciousness. I grew up beneath its shadow like a child raised under the threat of the mushroom cloud. You might say I am a member of the first HIV/AIDS generation. I've never known a world without it. This film is an account of my journey through the shifting sand surrounding HIV/AIDS. AIDS has been front-page news for nearly 30 years. Yet how much do any of us really know about HIV and AIDS? What is the difference between HIV and AIDS?
I don't know exactly.
HIV is wow.
HIV 是它的病毒，那 AIDS 是那个病，所以两种是完全不一样的东西。
It's the same
AIDS
thing.
is the actual disease.
It's the same
I don't know
thing.
really. I don't know the difference. I know HIV is less deadly.
Pour moi, il y en a pas forcément. Enfin, c'est vrai que j'établis pas de différence entre les deux.
What would you say is the difference between HIV and AIDS?
It's not a big difference, is it?
HIV/AIDS is just the starting point.
HIV, it's a virus. The actual virus is AIDS.
People around the globe were just as confused as I was. So I sought out the world's leading HIV/AIDS authorities, among whom were the discoverers of HIV, the key White House advisor on AIDS issues, and the executive director of UNAIDS' global response to the epidemic. Meeting with these distinguished experts, I candidly asked, what is the difference between HIV and AIDS?
The difference between HIV and AIDS is a really critical concept, and unfortunately it's one that seems to escape a lot of people, or just they don't remember it after hearing it.
HIV is a virus. AIDS is a syndrome caused by infection with the virus. So you don't get infected with AIDS, you get infected with HIV, and that causes AIDS.
The biggest problem with the HIV theory of AIDS is HIV.
There is a group of AIDS denialists that say that HIV does not exist and has never been isolated, which is as bizarre as it gets.
We do not say that HIV doesn't exist. What we say is that the presently available data does not prove the existence of HIV.
The reality is that HIV does exist and does cause AIDS.
I mean, the evidence is incontrovertible. HIV causes AIDS.
All right, that's a theory that's there. Let that theory be there. But let's have some other conversations. Let's have some other research. Let's have some other funding. Maybe something else is working here. No.
Why nearly 3 decades since its discovery does there continue to be debate over HIV? Why is there no cure in sight? To answer these questions, I needed context. The past is prologue, so my journey begins with a step back in time.
The National Center for Disease Control is reporting more cases of 2 rare and deadly diseases found in homosexual men. There is no apparent explanation for the outbreak.
Obviously, this is an issue with great emotional fervor, but...
How can we stay unemotional when people are dying every day from a disease the CDC has yet to name? For crying out loud. If the CDC won't name it, at least demand the press stop calling it GRID.
Well, unfortunately, I have to take credit for coining the term GRID, which stood for gay-related immune deficiency. We were seeing a cluster of gay men who were suddenly critically ill of pneumocystis pneumonia, which was the indicator disease of something new, and reported our findings to the CDC.
I was the chief of the STD division at the CDC at that time, when the draft report of 5 cases of pneumocystis in gay men came across my desk for review. Shortly afterward, cases of a very rare cancer called Kaposi sarcoma were diagnosed in young gay men. My first reaction was, this is an extraordinarily important finding.
The CDC was looking for something like that when it came along. They were looking for it already. They were hoping there was going to be a new plague because polio was over. The CDC's budget was getting decreased. This is back in like the early 80s.
There was double-digit inflation, a very high unemployment, a rapid military buildup, and a threat to decrease all domestic programs. And this led to reductions in force in the public health service, particularly the CDC.
The Center of Disease Control, CDC, in Atlanta was under threat for reduction and even theoretically for closure.
There were memos around the CDC that says, we need to find a new plague.
For them to justify their expenses and their existence and make their careers, they have to find infectious diseases.
We need to find something that'll scare the American people so they will only give us more money.
Once people recognized that this was likely caused by a virus, the media attention went from no news coverage to the most covered news story in history. People went from neglecting it to fear and panic. Maybe I can get it.
All of a sudden, AIDS was a very fundable project, and I suppose the psychology they worked on was the fact that they thought, well, in Congress, essentially this is white, straight, heterosexual men who are the congressmen, and if they feel they can't fuck around without being worried about AIDS, they're going to let the dollars out. And it worked.
Suddenly there was a lot of money available for anybody who wanted to study HIV. And nobody ever looked back and said, why do we want to study HIV? Bob Gallo said on television, causes AIDS.
The evidence shows that this disease is not merely confined to the gay community. I motion to call the disease acquired immune deficiency syndrome, AIDS.
In 1982, Dr. Harry Haverkos was one of the Centers for Disease Control's epidemiologists tasked with defining AIDS.
AIDS refers to a syndrome, and its definition changes periodically.
AIDS is a chronic disease. It's based on its immunodeficiency.
AIDS is not a disease, right? AIDS is a whole lot of different things. Depends on what country you're in.
When your CD4 count falls below a certain arbitrary level, by definition, you have AIDS.
You know, when someone's count goes down and then it goes back up again, it's, politically they may still have AIDS. Medically, I don't think they do.
If you develop any of a number of opportunistic infections or diseases, that puts you in the category of AIDS.
We don't even know what AIDS is. AIDS is so hard to define because they change the definition of it every year.
The definition of AIDS has broadened over time. It was revised in 1985, and then again in 1987.
The changes in the definition have been political. Every time they change the definition, the numbers go up.
The definition has changed many times. The biggest change was probably in 1993, which they then, added the CD4 count and HIV, and, so you could not even be ill, but if you had a CD4 count consistently below 200, you now had AIDS.
A closer look at the Centers for Disease Control's documents reveals that AIDS numbers actually declined in 1993, but a retroactive definition change increased the estimates by more than 100%.
The more diseases they could lump into this AIDS syndrome, S stands for syndrome, the better the chances are they get patients under that umbrella, the more patients they could catch.
As time goes along, definitions get used for a variety of issues, and some of those are not based solely on scientific decisions, but politics and capitalism and reimbursement comes into play.
For example, a person with hepatitis C, even say here in San Francisco, if you've got hepatitis C and only hepatitis C, you're shit out of luck.
Having an AIDS diagnosis, I get a free apartment. The city of San Diego pays my apartment.
I can have the state of California pay for many medications related to HIV.
I get Social Security benefits.
I can get discounts on my supplements at the local health food store.
I also get food stamps and in-home supportive cleaning services.
So I was basically a healthy person walking around, and I had all these wonderful little perks, You
get all these benefits, that we fought for and got. But the end result has been a sort of an imbalance. We succeeded. I'm glad we did, but it is a little unfair.
Politics, insurance, capitalism, benefits, you can be sick or healthy. I never would have thought that AIDS was so convoluted.
Right. Well, as I said, that has changed. How you define that scientifically has changed quite often. Which just makes it difficult to, for the lay people to understand. It makes it difficult for me to interpret the numbers.
Africa is purported to have the highest incidence of AIDS cases on the planet. So I trekked to South Africa to witness firsthand the impact of AIDS on that troubled continent. Around 10 million of South Africa's 48 million people have been reported to have AIDS. It's only a 10-minute drive from Cape Town's pristine modern airport to the squalor of neighborhoods said to be ravaged by AIDS.
All the facts on HIV and AIDS with Griselda on Metro FM Talk.
It's World AIDS Day. There are functions, there are gatherings. The international theme is Stop AIDS, Stop HIV and AIDS. Keep the promise. So many years later, we keep saying the same things, HIV, HIV, HIV. And yet
each time I hear words like HIV and AIDS, I just want to pull my hair out. I'm just so fatigued about how we've packaged the messaging. All we talk about is AIDS. It's a sex virus. You have to use condoms or you die. It's a sick and sad message. People can't think outside AIDS anymore. It's just a shocking, sad reality.
The first AIDS meeting on the continent of Africa was in 1985 in Bangui.
We were there with a few people who had experience on AIDS in Africa. And one of our problems was, how can you diagnose AIDS in Africa in the absence of very sophisticated laboratory support?
Even though by 1985 there was a HIV test, most of Africa didn't have access to it. So one of the things that we did in that meeting was to sit down and hash out the so-called Bangui criteria for the diagnosis of AIDS in Africa. The idea was, what would be a simple way for a clinician to look at a patient and say that this patient likely has AIDS?
We say somebody who has a combination of certain signs and symptoms like major weight loss. And if you have a combination of that, you can say this is probably somebody with AIDS.
They wanted a clinical case definition where they could decide that someone had AIDS just by looking at weight loss and persistent fever and so on.
It gave something to clinicians in Africa to diagnose AIDS.
And that helped in the overall effort to count cases, because we needed to know what was the impact of the epidemic.
They could discover AIDS all over Africa at that point. They could say that we are all at risk, but they could say it's spreading around the world. They could say it affects women as much as men, because almost anyone in an African hospital could be diagnosed with AIDS without having to do the HIV test at all.
Whole nations have been led to believe that, in some instances, that they've got large percentages of their population infected and doomed because of this supposed sexually transmitted virus. It's such a tragedy.
Daniel, a lot of people here is very sick and is very dying.
What kind of sickness do you see around here?
It's HIV/AIDS.
What is AIDS?
We don't know. We don't know.
So here you're living in a mud hut, and here some kind of white men with doctors who you respect, and they tell you that there is now among you an invisible disease, and it gets into your blood and can stay there unseen for years, and when it manifests itself, it's going to manifest itself in the forms of disease that you've always known.
Maybe if you look skinny, if you lost weight, maybe they'll simply say hey, you have AIDS, or you're coughing a lot, maybe they'll say you have the disease.
This cannot help but create extraordinary paranoia in people's minds. They say, well, what is going on with us? My neighbor next door he's got malaria. Does that mean that he's actually got this dreadful disease that the whites are talking about?
Like I could be fat now, then the mentality is that if ever I become teen, then it could be one of the symptoms. I have to ask myself, why now? Why at this time?
Dr. Christian Fiala argues that many doctors have misused the Bangui definition.
In the era before AIDS, we had to admit we don't know the diagnosis and we could hypothesize. But nowadays, what doctors do is, well, if we don't know what it is, it must be AIDS.
We did have patients with the conditions we now regard as AIDS defining, even before the advent of AIDS.
People could have TB and not have HIV and fulfill the Bangui criteria. They'd lose weight, they'd have TB, and they could look like they have AIDS when they don't.
Is this Bangui definition still being used today?
I'm fairly certain that in many parts of Africa where there's still little or no testing available, that definition is still used, and I wouldn't be surprised that it's used in the poorer parts of Asia.
This word AIDS, I don't know what it is anymore. I don't know what we're talking about anymore when we talk about AIDS. AIDS is one thing in Greenwich Village and a very different thing in Kampala, Uganda.
I visited the World Health Organization's website searching for answers and discovered there are currently more than 12 different definitions of AIDS worldwide. So I turned to Dr. James Chin, former head of the WHO's Global HIV Statistics Unit, for an explanation.
Some countries, they felt they were a little more sophisticated than in others. And you have, along with the epidemic of HIV, epidemics of HIV/AIDS experts. And some of them will not necessarily adhere to any international definition. They'll make up their own definition.
High school biology class taught me that diseases and syndromes cannot differ from country to country like languages. It was becoming clear that HIV and AIDS were distinct, separate entities, and that AIDS was diverting my attention from the real culprit, HIV. Where to next? The place millions have had their lives changed forever.
She says, we have your test result, you need to come in and get it. And I was like, tell me now, Cheryl.
she was like, Kim, we really need you to come in and tell me. I said, then I know it's positive, Charlotte. You would just tell me over the phone. And she's like, Kim, don't panic. You can still have a normal life.
I can still remember his face. I can remember his eyes. And all he said was, I'm so sorry. I think you should put your affairs in order, and you might have 5 years.
I said, Cheryl, I have to get off the phone right now. I have to go tell my dad. He started crying. This isn't the way it happens in the movies.
It's 7 a.m. here in Johannesburg, South Africa, and I'm a little nervous because I'm about to go in for my first HIV test. Have you ever gone in for an HIV test?
No.
No. No.
No.
Yes, I have actually, a few years ago.
Were you nervous, apprehensive?
I was, very frightened.
The nearest testing center wasn't in a hospital or in a doctor's office, but on the main concourse of a bustling train station, beneath a few portable tents. HIV testing facilities are everywhere, from street corner kiosks to shopping malls. I'll be tested with a rapid test, which looks for antibodies to HIV.
So, have you ever tested for HIV before?
No.
Except no.
Never.
It's the first time.
First time.
Wow.
Is this your questionnaire?
Yes.
Okay.
These tests claim to be HIV tests. I'm going to read from a section that says limitations of the test. The specificity of the Reveal Rapid HIV Antibody Test for blood specimens in low-risk populations has not been evaluated. They don't know, in their terms even, how well this test is going to work in people they don't want it to work in. Low risk. We don't think you're at risk.
Tell me about your sex life.
My sex life?
Us? Since you haven't exposed yourself, lied, sex, you're not homosexual, you're gay, you know your story.
Yes.
Let us go and find out.
In 1990, we flew to Romania to adopt a baby. We found Lindsay, and she was only 2 weeks old. I can still remember that feeling, just holding Lindsay for the first time, thinking, now my dream is going to come true. I'll have one of those children. Before we left Romania, we had to make sure that she didn't have HIV. We had to find a doctor, and he did the test, and it came back negative. We flew home just after Christmas, 1990, and thought we had it made.
So I'm going to do a test, which comprises of 2 test kits. These 2 test kits are from 2 different countries. Why you are putting them being 2, you want them to confirm each other. Can they be 2 different outcomes? Then this one is a determiner, which is going to give us the final results.
Can I just ask you something, as someone who's a little nervous?
Okay.
It seems like if this is positive and this is negative, my life hangs in the balance on whatever this one is.
Yes.
But how do we know that this one's accurate when both of these were inaccurate?
This one has been tested to be the one that is going to take out the results that are correct.
Oh, so this one is more accurate than these 2?
Yes, according to what we have learned.
So why don't we just use the more accurate one to begin with?
Well, you know what? What if now the more accurate one has a discrepancy? How are you going to find out?
I don't know.
Rapid tests in Germany, it's not allowed for standard diagnostics.
May I ask why? How come you don't use rapid tests for standard diagnostics?
Several professional organizations who decide as an expert committee on guidelines how to do things. None of these responsible societies recommend it for scientific reasons.
We always say to our clients, even if you have tested here, you can go to other centers and go and test and verify your test. You cannot say you're 100% because you find clients going from area to area doing tests. And they come with stories that I was negative at a certain area, I'm positive with you.
How do they decide whether they're positive or negative?
We cannot tell because we are using a rapid test.
It occurred to me that perhaps the HIV epidemic is reported to be so widespread in South Africa and other poor nations simply because they use these inaccurate tests.
There's the saying that if you knew how what sausages are made of, most people would hesitate to sort of eat them because they wouldn't like what's in it. And if you knew how HIV/AIDS numbers are cooked or made up, you would use them with extreme caution.
I decided to investigate HIV testing protocols used throughout the developed world.
When we're testing people for HIV, the first thing we do is a screening test, and it's usually a test called the ELISA.
But there are also now available rapid assays that can be used as screening methods.
Because they're faster. And we all know faster and cheaper is more efficient.
If an ELISA is positive, it does not mean that the patient is HIV positive. That's the problem.
If we're using antibodies as a screening test to tell who's infected or not, very occasionally you can get false positives.
So screening tests by themselves should not be used as a definitive measure of infection. That's why we use a screening test to pick up all the cases, but we use a confirmatory test to eliminate any false positives.
Take it easy. I'll just get the site.
It should be emphasized that most of the developing world uses only screening tests to confirm an HIV diagnosis. There are no confirmatory tests.
Time now is 25 past. At 22, the results will be out, which is going to be 15 minutes.
9 days after returning home, Steve and Cheryl's pediatrician ran a battery of tests on Lindsay, including an HIV test, even though Lindsay had tested negative for the virus in Romania.
Dr. McHugh called us and said, we run into some problems with the testing that you did at Methodist Hospital, and you'll need to come right in and see me.
I said, well, what is it? She says, well, I'm not going to tell you over the phone. I said, I need to know exactly what this is. He said, we got bad news that she tests positive.
And he said she'll have a 20% chance of living to age 2. It was just a shock. Just a shock after all this joy and happiness. We finally found our daughter, and I'm dancing around Romania, and now I come home, and it's like somebody could just stab me. And then I had to call my mom, and that was the worst phone call I've ever had to make, because I even remember saying, that poor girl, she's just not going to make it.
So that we don't have to go to somebody and say, well, you might be infected, but it might be a false positive. We do a second test. That's a test usually called the Western blot.
In 1992, when I was told by my doctor that I was HIV positive, that was only a verbal admission to me. She didn't give me the written paper that came from the lab that tested my blood. I found out that it says this indicates possible infection by virus.
There can be mistakes from the antibody test, and there are conditions that can cause the test to be inaccurate.
Now that I've got the package insert for that test kit, it says positive results using any specimen type should be followed with additional testing. But this is the test that they use to confirm with.
This has a margin of error done properly that's extremely low. In other words, it's one of medicine's better tests.
I don't think the Western blot is a useful diagnostic test. I don't think it's worth doing.
Did he give a reason? Anybody can say anything. I think it's stupid to drive a car. But come on, you got to give a reason.
It's a useful prognostic test. Once you know that someone is infected, then you can follow their antibody response as well with Western blots.
I'd say he's absolutely wrong. It has a complete usefulness.
You don't need a Western blot. And it's become a dogma in HIV research that you need one ELISA followed by a Western. You don't. You need 2 different kinds of ELISAs made in 2 different formats.
Would you ever want to confirm somebody is positive using just ELISAs?
No, never. It's against the rules. It's against the recommendations.
It's a turbulent sea of argument about how can we use this test? When can we use this test? Why does this test have no standard?
We have a group now of about 40 patients that have no detectable virus.
in their body, but they're not being treated. So the first question is, are they really infected?
So the Western blots can have false positives?
No, the Western blot was negative too. But they were told they were positive by a lab, yes, that misread the Western blot.
Heavyweight champion Tommy Morrison tested positive in 1996. 11 years later, in 2007, he tested negative multiple times, allowing him to return to the ring.
There's constant discussion in the community of people who do diagnostic testing and the blood bankers about how to read these tests.
When you're looking at this Western blot, how do you determine what is a positive?
You need a certain number of bands being present. It depends a little bit on the producer of the test.
It depends on the manufacturer. Is there different criteria for what might be a positive?
Yeah, there are different criteria from the manufacturer. Thank you for the word. And also there are guidelines from the WHO and UNAIDS.
Well, HIV infection is diagnosed with rather now routine laboratory tests, which for which there are criteria for diagnosis established by the manufacturer, FDA.
Claudia showed me the package insert that comes with the Western blot. It contains 8 different sets of criteria for diagnosing HIV infection.
Because of the different criteria that apply in different countries, you can be considered, you can test HIV positive in one country and be given an AIDS diagnosis as a result of that, whereas in another country you won't test HIV positive and you won't be given an AIDS diagnosis.
It's ludicrous that you can be positive in one country and not positive in another.
Theoretically, I could be diagnosed with AIDS in the United States, but if I take 3 steps to my right, I wouldn't be diagnosed with AIDS, or I would lose my AIDS diagnosis when I crossed the border.
In 1992, I was encouraged by a doctor to take what's called an HIV test as a matter of social responsibility, and I was shocked and devastated and horrified when the results came back positive. It was one of those moments that everyone fears their whole life. A week later, I take the same test to an AIDS specialist. He looks and says, "This isn't a positive test. I don't know what this test means."
Since a false positive looks like a true positive, how can you ever distinguish whether it's truly a positive or a negative?
Well, that's a great question. It's going to be very hard to determine a false positive.
So I take the test again, and this time my results come back marked from the lab, indeterminate. I'm faced with the decision, do I want to wait 6 weeks to test again or do it right away? I opted for right away. My results that time come back positive. Took it again, came back negative. I took it again, positive.
What happens if you're positive on one criteria but negative on another manufacturer's criteria? How do you decide who's infected and who's not?
You will use the most sensitive criterion.
In late December of 2007, I read about new legislation passed in New Jersey calling for the mandatory testing of pregnant women or newborn infants should the mother's status be unknown.
HIV mandatory testing, to me, is a no-brainer.
I'm very much opposed to the concept of mandatory testing of any population because the tests are scientifically shown to be unreliable and inaccurate.
You have no reason to fear this bill. And my hope is that eventually this will become a federal law so that every woman in this country would be tested.
But HIV testing isn't an absolutely precise science.
When I confronted my doctor about that, she said, we're way past Western blot now. We have the viral load test. But when you get the package insert for the viral load test, it says,
if you test positive, you are considered confirmed infected with HIV. But at the bottom of the page, in fine print, it states a person should have additional testing.
It does not allow you to tell a single person on this planet that they are HIV positive. And it's a scandal that this test continues to be used.
So again, I'm asking, where's the test? Where's the test that can confirm a diagnosis of HIV infection? And I can't find one.
I have the package insert from the manufacturer which they supplied me with. And under limitations of the test, it states that risk factors should be used in conjunction with the test. Has a person had sex? Have they used drugs? Have they had a blood transfusion? And then in conjunction with the test, not the test alone, but with the test, then you decide whether the person is positive or negative. Do the answers to these questions help aid in the diagnosis?
Oh, of course they did.
Really?
They do.
Now, if I tell you that the test you took was lousy and didn't mean a thing, does that make any difference for everybody to hear?
It'd make a difference for me.
Yeah, I know.
How can we say that HIV is the cause of AIDS when we don't know, based on current tests, whether or not anybody diagnosed positive actually has HIV?
President Thabo Mbeki will officially open the AIDS conference tonight. Delegates here are hoping he will finally separate himself from the AIDS denialists.
We remain convinced of the need for us better to understand what would constitute a comprehensive response in a context such as ours, which is characterized by high levels of poverty and disease. As I listened and heard the whole story told about our own country, it seemed to me that we could not blame everything on a single virus.
I thought this man must be an idiot. Everyone in Africa is dying of AIDS. I know this because I read the New York Times. It's beyond doubt.
Rion Milan was hired by Rolling Stone to investigate and debunk President Mbeki's misguided ideas. Where to begin? The numbers.
My very first action, I opened Johannesburg's Yellow Pages. I thought that I could illustrate this with a scene that began in a coffin factory in Johannesburg, where workers are working overtime trying to create caskets for these mountains of people who are dying of this condition. And I discover that in the midst of what UNAIDS says is a plague, is that half of the coffin factories in Johannesburg have gone bankrupt.
In South Africa alone, it's about 1,000 people dying every day from AIDS.
If you're an advocacy agency and you perceive low numbers to be bad, your bias may be to accept higher numbers even if they're not scientifically sound.
I know Jim Chin quite well, and I was the chairman of the steering committee on epidemiology of the global program on AIDS in WHO when Jim Chin was in charge of epidemiologic estimates. And we could never get information how the WHO estimates were made then. So we were very critical in these days because we felt it was not based on enough evidence.
It's possible that he didn't read the materials we sent him or he didn't understand them, but we did send information to anybody that wanted to know about the estimates because they were pretty transparent.
In an attempt to get to the bottom of the statistics debate, I've come to Geneva, Switzerland to look at the World Health Organization's official numbers. And what we found, there are no numbers, only assumptions and estimates.
How are the monies divvied out to states for AIDS prevention or AIDS treatment? How is government monies sent out to different states and communities?
The more AIDS you have, the more money you get.
Exactly.
The AIDS buenas have a vested interest in maximizing and squeezing the data to get the worst possible scenario that they can out of it, because the worse the situation is, the more compelling their fundraising claims are.
When UNAIDS was created, about $250 million was spent on AIDS in poor countries. 10 years later, it's $10 billion. That's an unprecedented increase. Still not enough.
When that was created, the first thing Peter Piot said was that UNAIDS is an advocacy agency, pure and simple.
And this was my first objective when I came into this job, and that was put it on the political agenda.
He divested himself of all of the program aspects and the scientific aspects of AIDS.
This is not a scientific issue. This is a matter of politics.
Except one unit. He kept sort of the numbers unit.
we are really doing a major disservice to say it is not as bad as it looks like, because actually it is much worse.
1 month after my interview with Dr. Piot, the Indian government slashed their estimates by nearly 60%. Shortly thereafter, UNAIDS acknowledged they'd been overestimating global HIV statistics for more than a decade.
They've painted themselves into a corner, and now their house of numbers is falling apart.
America is leading the fight against disease, and I call on you to double our initial commitment to fighting HIV/AIDS by approving an additional $30 billion over the next 5 years.
Noble corruption, misuse of statistics, in order to convince people that there's one hell of a problem out there, guys, and we've got to go and do something about it.
If you look at the real world, you know, how many people are infected, diagnosed to be infected, and eligible for treatment? There are very few compared to the number.
In April of 2008, Congress approved a $50 billion expenditure for AIDS treatment and prevention.
The vast majority of the world's population is not at any measurable risk of HIV infection.
No measurable risk. Growing up in the age of AIDS, I was taught there were 3 certainties in life: death, taxes, and contracting HIV from unprotected sex.
If you don't use a condom, there's a lot of chances that you can actually get the killer disease that is AIDS.
I did a study of the heterosexual transmission of HIV in California, and we recruited individuals who were infected with HIV. Then we recruited their sexual partners, and we looked at whether transmission, in fact, had occurred.
Padian runs a study. It's a 10-year study with the world's most virulent, terrifying sexually... I mean, this thing jumps, off of penises into vaginas miles away. How many of them do you think, after 10 years, with the world's most terrifying, virulent, sexually transmitted disease, came up positive? Nobody. Nobody. Nobody who was negative came up positive. 0.
I think HIV is more difficult to transmit than probably most other sexually transmitted diseases. I mean, I think that's pretty widely known.
If I were to have unprotected sex with somebody who is HIV positive, how many acts would I have to engage in before I got the virus?
Just one. I think one would be enough.
First act?
One is enough.
Remarkably, HIV is a difficult infection to transmit.
This contradicts everything I was ever taught about the sexual transmission of HIV.
AIDS is the best example of what's really scary and alarming and dangerous about our culture right now, which is that it's a culture of PR. It's a public relations phenomenon. The truth doesn't matter. What matters is the image. If we were talking about reality, the reality is that AIDS is over.
Somebody decided in the early 80s that there's this infection called HIV. And upon deciding that, I don't think it was debated enough.
In 1983, Dr. Luc Montagnier and his team of researchers identified what they thought might be the cause of AIDS.
I really was excited because we knew it was a new type of virus, not shown before in men, and very likely to be the cause of AIDS. Of course, at that time we didn't have the full proof of what's the cause of AIDS.
That initial work was rapidly reinforced by additional virology and serology studies in Bob Gallo's lab in Washington.
Gallo got pushed by the Reagan administration that wanted to do something on AIDS finally. They literally told us to just close CDC's lab down. We don't care about it. There's a bunch of gays, who gives a shit? And that was really their whole story. So the whole thing that they rested their political response was, well, we discovered the virus.
Good afternoon, ladies and gentlemen. The probable cause of AIDS has been found.
Credit must go to our
eminent Dr. Robert Gallo, who directed the research that produced this discovery.
What was new that day is for the first time we were saying, that's the cause, I'm sure.
It was a silly press conference. There was not evidence then that HIV was the cause of AIDS. It wasn't called HIV. There was certainly evidence that he didn't discover it. It was discovered in France.
The conference was held before any of Robert Gallo's papers were published, therefore before any other scientists had a chance to review them and look at the evidence and see if he got it right or wrong.
Gallo's philosophy was to have people to whom he would give the virus be in his own control, so that any information that came out of that would come through him, so that he got all the information, indeed often put his name on publications and would, quote, collaborate with them. But should you have any broader view other than his personal glory and your personal glory, this was not a scientific pursuit in any way.
The U.S. Department of Health and Human Services decided from now on we are only going to fund AIDS research that assumes that Robert Gallo's virus is the cause.
I didn't think that HIV in 1984 was the cause of AIDS. That's why I did the study comparing gay men with Kaposi's and gay men with pneumocystis. I assume there must be something else. If you go read my paper from 1985 that Kern and Jaffe would not sign on to, I actually lay out the cofactor hypothesis in that paper.
I'm a promoter of the role of cofactors in AIDS.
Well, cofactors just says that the cause of a disease is by more than one factor.
Just simply being infected with HIV is not going to do it. You need certain cofactors.
Cofactors are not necessary.
Dr. Fauci would say HIV causes AIDS without the need for anything else. That's kind of ridiculous.
The data that indicate that any different type of infection like mycoplasma or something like that is a necessary cofactor, I believe those theories have been debunked.
What the fuck does he mean? Sorry. What's it? What does he mean that there are no cofactors? Where's he coming from? There's cofactors for everything.
Cofactor implies something specific. And it really gets us off into tracks that are wandering.
Gallo isn't gonna change his mind. But he's probably 70 years plus now. He's going to remember things. You know, we all remember things that are good for us, and we forget the bad things. The cofactors are important to really understand how people get ill, why they get ill.
What is asked of an AIDS journalist is to deny an existing reality, which is a strong, growing body of dissent on a scientific question.
In August of '92, my dad had just read his latest National Review, and there was an article in there about Peter Duesberg, a maverick cell biologist. He said he didn't think HIV caused AIDS.
To deny that this well-identified, well-characterized virus is linked with AIDS is, to my mind, just potty.
He said, I can't replicate this in my office. I can't get this virus to do anything. We thought, oh my gosh, my dad is really daydreaming. He's trying to wish this all away because he knows how upset we are. Just wish it all away, and everybody will be gone happy.
If Fauci would say, here's $1 billion for alternative theories of AIDS, you wouldn't believe what's going to happen. A lot of HIV researchers overnight would would start cofactors. The first year they would call them cofactors of HIV. And the next year, the co would be topped, the HIV would be topped the year later.
Peter is highly intelligent. He did excellent work. I mean, no wonder he got a full professorship in Berkeley and was nominated to the American Academy of Sciences. But I told you very frankly that he's killing people with his theories, and I still stick to that.
They attack him, they attack his ideas, and they present some bogus way of refuting what his science has said that doesn't really refute it.
Peter Duesberg? Yes. They are all prostitutes, most of them, my colleagues, to some degree, myself. You have to be a prostitute to get money for your research. You're trained a little bit to be a prostitute. But some go all the way.
In light of all this scientific uncertainty, I asked Dr. Fauci for evidence linking HIV to immune deficiency disease.
When you put the combined findings of the initial characterization as a distinct retrovirus isolated by Montagnier and his group, together with Gallo linking the virus to being the cause of AIDS, and they put those things together, and that's how we have a confirmation of the causative agent of AIDS, namely HIV.
Still unclear about the evidence for HIV's existence, I decided the best way to verify it would be to actually see it. I asked Dr. Hans Gelderblum, a world-renowned electron microscopist, if he thought there was any reason to question Dr. Montagnier's published images.
I've seen these publications, stamp-sized images. It's a nuisance. It's a nuisance. You do not really see much.
When we saw that photo, we said, suggestive, but not convincing.
Dr. Gallo, 1 year later, published photographs he claimed to be of HIV. Were his any better?
These pictures were not so impressive. They were not much better than Montagnier's images.
It's one thing to look like, and another thing is to be a virus.
In 2002, I stumbled across an article by Valander Turner and Andrew McIntyre of the Perth Group in Australia, and it questioned whether there's ever even been found a virus. I became consumed with researching this. I could read from morning till night every day, and every link to another link, and I would email to these people and say, where's the test? I want to know. Am I dying? Am I contagious? And they weren't even very kind. They were just like, read our articles again. How many times do we have to tell you there's no test?
It is crucial to understand that an AIDS diagnosis is forever. It cannot be reversed or alleviated.
The stigma attached to people knowing that you have it and are living with it is worse than actually living with it.
I can't think of anybody who's ever been evicted from their apartment because they had breast cancer or because they had cerebral palsy.
I did a campaign to address stigma. The message is that if anyone is infected, we are all affected. Some have it medically, some have it socially, some have it culturally. And at the end of the day, if it exists anywhere, it exists everywhere.
We don't all have AIDS. And once you start bullshitting, it gets a problem. You know, we don't all have AIDS. We all have to be sympathetic to AIDS. And yes, there's all kinds of people who get HIV infection. But you know, we don't need to make, in my view, non-truths, you know, or just to have a slogan or a symbol. We all have AIDS. No, we don't all have AIDS.
But my message is you do have it, whether you want to accept it or not. Are you medically susceptible? Maybe not. But are you socially vulnerable? Yes.
In June of 2007, the BBC featured a news story which began, HIV infection theory challenged.
Living cells are complicated, and how they work inside the body is even more complicated. So there's still a lot of debate as to how exactly HIV causes AIDS.
In March of 2008, the Washington Post went on to state that multiple surprises have reminded researchers how much they still don't know about HIV's biology.
HIV has got to get inside the circulation of the body, and it does that in ways that are not completely understood.
The prime target for HIV is a T cell population called CD4 helper T cells.
The way that the virus gets into the target cell, it fuses its membrane to the membrane of the cell.
I don't understand the fusion process. I don't think anybody completely understands it.
We have a relatively poor understanding of how viral proteins interact with proteins in the cell.
How come our antibodies aren't able to keep HIV in check?
It's an excellent question. That's one of the great stumbling blocks.
They can't prove that HIV is the cause of AIDS. Okay? They cannot prove that HIV is the cause of the collapse of the immune system, no matter how many scientific journals says it is. When you go to the basic research, it doesn't prove it.
We are almost convinced that there is other factors that are involved in the loss of CD4 cells, and we don't know yet all the mechanism.
How HIV depletes the T cells so an individual advances to AIDS is probably due to multifactorial elements. One is it will kill the cell eventually that it infects. HIV does not necessarily kill the cells it infects. Some T cells are directly killed by HIV, and other T cells keep the virus in check. It's a silent state within the cell. And I think in many cases these cells can return to a normal function.
Can that cell return to a normal state? I don't think so.
When I was told I was HIV positive, I accepted that on a very deep level. But only by having the courage, the open minds, and the open hearts to answer these questions are we ever going to know how to help people, how to do what we need to do to help people.
In late 2007, Science Daily reported that 3 prominent research teams had published papers in the Journal of Immunology challenging the theory that the sudden loss of T cells triggers disease and AIDS.
The details of HIV pathogenesis, how HIV kills people, are still being worked out.
If the sudden loss of T cells in HIV-positive individuals can't explain why people get disease, then there must be cofactors that cause people to get sick and die, or factors that have absolutely nothing to do with HIV. While researching HIV hotspots, I began to realize there is a direct correlation in these places with another condition that leads to immune deficiency and death.
I think it is important to keep in mind, especially for us in the West, that poverty is not a romantic issue. It is a deadly issue. Poverty leads to diseases and premature death, period.
When you look at the symptoms that they talk about, for people that are HIV positive, you find that some of them, they are more related to malnutrition.
Their nutrition is not very equilibrated. They are in oxidative stress, even they are not infected with HIV. So their immune system doesn't work well already.
People are hungry, they're underdeveloped, there are no hospitals, there's no proper medical care. You take away poverty, you're giving people an ability to fight infections.
These are toilets.
Yeah. We don't have flushing toilets. When flies get into the toilets, they actually come back to touch our food, you see. And look, we need to take that food. And we don't know which infections are there.
So how do you expect people must survive in these places? Look there. How many white people they are staying in this house like this one? In Cape Town is different. You go to suburbs, you see the suburbs is different from our places.
Why have we done nothing for African people, just like give them clean water so they don't die in infancy of diarrheal disease and stuff like this? All we care about is HIV, AIDS. Well, question. Well, there's money in it. There has to be, other dynamic working to lead public interest to the African situation and ignore clean water, sanitation, malaria. I mean, things that kill people.
This is the beginning of a war. It is a war to reclaim our health.
In 2008, USA Today published a news story that stated, if we look at the data objectively, we are spending too much on AIDS. About $10 billion a year is spent on AIDS, while 2 billion people live with no sanitation. 1 billion lack access to clean water. And malnutrition kills someone every 10 seconds. These factors enable diseases to thrive and severely weaken the immune system of those living in such squalor.
If we were to take all that money and put it towards developing poor countries, God would be so proud of us, indeed, because we would have taken away the major challenge that's facing humankind, and that is people dying in silence. It's slow, painful deaths from being scared of something that was just packaged as AIDS.
Could it be that the real epidemic is extreme poverty, not HIV? On the other hand, HIV allegedly occurs in the United States as well. So I looked for alternative causes of immunodeficiency in this wealthiest of nations.
There are other ways you could produce a condition that looks like AIDS, but they too will be some source that causes a severe defect in the immune response.
19 million Americans now, 19 million are taking illicit drugs every day. But we don't talk about this. This is politically incorrect.
There have been a number of theories as to what the origin of HIV/AIDS is. One of them was a theory that certainly turned out to be completely incorrect, that it's a lifestyle phenomenon.
There's a large epidemic of STDs in general in the late 70s, and particularly in gay men. In San Francisco, New York, Los Angeles were meccas of the new lifestyle, which came from the liberation in the post-Stonewall era.
The more partners you could have, the more you were striking a blow for gay lib. I remember talking to one of the people who was at the forefront of that lifestyle. I very much felt like if he did have another bout of syphilis or gonorrhea, whatever it might be, that it was like a notch, a victory notched up for his right to exist as a gay man.
I went on a vacation to Los Angeles, and it wasn't that wild, but one night I went to a bathhouse. I came home with, get this, at the same time, syphilis, gonorrhea, and 2 forms of parasites.
I have no views about that, you know, in a judgmental kind of sense, but certainly from a public health point of view, that's kind of a prescription for disaster, because AIDS first occurred in these men who were not healthy for reasons that were obvious to everyone.
You're talking thousands of partners, sometimes hundreds, per month, lots of antibiotic use, lots of drug use, and the result were people were getting pretty sick.
There's a drug called amyl nitrate that was developed in the 1850s and 60s. Came in ampules, and they became known as poppers because you'd pop them when you open these ampules up to sniff them when you had coronary artery disease. The first AIDS cases, for example, that Mike Gottlieb reported were all 5 gay men. They were young. They all used poppers.
Poppers, something you walk around huffing all night. I mean, it says flammable, fatal if swallowed on the side of the bottle. They're walking around huffing it all night long. Why? Gives you a great rush.
Poppers was a sex drug. They were in every gay bathhouse, every bar, every porno bookstore across the nation. Poppers were visible on the dance floor in the discos. At the end of an evening, the bartender would announce last call for alcohol, last call for poppers.
It was like a mad, wonderful kind of a dance that was being done. But if you think that can happen forever, you're wrong.
Whether or not HIV exists, whether its role in a weakened immune system is almost irrelevant when you compare it to what was going on at that time.
The lifestyle explanation proved politically unacceptable, but the virus explanation proved very, very acceptable to many different parties.
Pneumocystis pneumonia and Kaposi's sarcoma were the hallmark diseases for AIDS in the early years.
To go back and deconstruct it and say, what exactly did cause pneumocystis carinea?
I remember the first patient that I ever saw, my resident brought me to see a young gay man with pneumocystis pneumonia. I knew a little bit about the use of poppers or amyl nitrite inhalants, and I started asking the patient if he used them, and it turned out that he was a very heavy user of amyl nitrites, and much to the surprise of my students, I said, I think the man probably has destroyed his pulmonary immune system by inhaling this toxin.
What exactly caused Kaposi sarcoma? We know that now. It was amyl nitrite.
We saw KS decline, interestingly parallel to the decline in popper use.
We now know that Kaposi sarcoma is caused by a second virus, the human herpes virus strain 8.
To rescue comes another virus. As always, when you're in trouble, you find another virus.
If one wants to look at really what causes this, we've got to look beyond just HIV and just beyond HHV8.
Calling it a disease, see? And to give it that name, AIDS. So everything's included under that, and you don't have to just say, I mean, if you just said, you know, these people are getting a lot of weird diseases, all kinds of diseases, it wouldn't have had the same impact. It was much better to say, there is a infectious organism on the loose in America, and it could get you.
I had interviewed the world's leading HIV experts and discovered that the two benchmark diseases of AIDS have alternate explanations. Once again, I turned to Dr. Gelderblom, seeking proof of HIV's existence in the most recent images available.
Here you do not see anything about the details, but I would say it's probably a virus.
These are HIV here?
Yeah.
Oh, so are these HIV too?
Yeah, yeah.
Everything's
Probably.
Probably.
Yeah.
What can I tell you? You know, I mean, it exists.
Gallo said he had all these viruses, and it was a lie.
I think HIV totally has turned out not to be the cause of AIDS. HIV has turned out not to be.
Gelderblom's images, said to come from isolated HIV cultures, provided no proof for HIV's existence. So I asked Nobel laureate Dr. David Baltimore and Dr. Robin Weiss how they would isolate and photograph this elusive virus.
Well, didn't Dr. Gallo do that? I mean, he actually isolated it, so... Yeah. I mean, why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook, you know? I got other things to do.
They're embarrassed. The scientists have been embarrassed about this. They know that it's flawed.
In 1987, the CDC made two mind-boggling changes in the definition of AIDS, which are in effect today. You can be diagnosed with AIDS without ever having an HIV test.
In 1987, I had a lesion on my arm, and it was KS. Kaposi sarcoma.
The doctor diagnosed you with AIDS without an HIV test?
Yeah.
You can be diagnosed with AIDS if you've tested negative for HIV.
Alvin Friedman Keene found 16 patients with Kaposi sarcoma among gay men in New York City in the 80s. They did not have HIV infection. Yet they had AIDS by our definition, right?
In a World Health Organization publication, Dr. Chin writes, "It should be emphasized that the surveillance definitions for AIDS were not intended to be reliable indicators of HIV infection."
If you have thousands of documented cases of AIDS without HIV, how can HIV cause AIDS?
Why do you believe that HIV does cause AIDS?
Because that's all the information that I've been given.
Because we've never been taught anything different. We have read it, heard it.
Because that's what the scientific community has told us.
Scientists are supposed to observe, experiment, and reason from what they observe. They're not supposed to grab hold of an idea and cling to it and adjust everything else in their perceptions to fit that idea.
I think an HIV positive test means that your life is forever changed. You have a whole new battery of things to consider for yourself. What does it mean to me?
It's very hard to find anyone who supports you when you say, I don't think I'm going to die of HIV or AIDS.
The typical model of HIV equals AIDS equals death. How invested am I going to be in that model?
Everyone who's infected with HIV would like to deny it. It's a bad prognosis. It means you're going to take drugs for the rest of your life, et cetera, et cetera. So there's people who want to say, I'm one of the people who tested positive, but I'm not going to get the disease.
Do I start treatment? Treatment meaning the antiviral drugs, if ever.
We started taking Lindsay to a doctor at the Children's Medical Clinic. She gave us a prescription for retrovir syrup, which is AZT. It was so important for us to get something to help our baby that we sat on the floor in the pharmacy and gave her first dose.
Shortly after Lindsay began AZT treatment, side effects began to emerge.
Her eating habits changed quite a bit. She didn't eat well. She was hard to handle at the table. And then the leg cramps started. Once that started, it got progressively worse.
She would just grab him and go, ow, you know, screaming in the middle of the night. Just like it was it wasn't an ache, it was like, must have been sharp pains. It's just made you feel sick to your stomach.
Any drug active on HIV will be toxic because it's not 100% specific on the HIV enzymes.
When we switched over to the university, then the dosage of AZT went up, and that's where she started flattening out on her growth chart. The doctors would try to put a positive spin on how well she was progressing. It was mainly in the T cells that weren't always a positive situation.
Yeah, the T cell count would go down, and then the doctors would say, well, maybe we better raise that AZT dosage, get that T cell count back up. We were going, I think it's kind of making her sick because she doesn't want to eat. She's having leg cramps. And they'd say, well, it's the HIV, and that's what it does. It's all part of the package.
The treatment causes a very similar condition we would expect from an AIDS patient. That's why nobody noticed that there was something wrong with the treatment.
I remember in 1992, after I first tested positive, I became involved in an organization called Women at Risk. There were 11 of us at the time on the board and involved in the group. All of us except 3 were on the medications. In the year and a half that I was involved with Women at Risk, every single woman in that organization on the drugs died. Every single one, except the 3 of us who weren't taking them.
We weren't just given handfuls of AZT, we demanded it.
AZT should be praised. Where is your humanity?
We considered the FDA not giving us these things as being anti-gay instead of being responsible. And so we went and we lobbied and we pushed for all these things, and we didn't think clearly about what it was we were asking for. It's like that saying, be careful what you ask for, it may come to pass.
That's the very reason why everybody believed HIV is a deadly virus, because the HIV-positive patients at that time got a deadly treatment.
Despite the billions spent on the drug, tens of thousands of people with AIDS have died. And now a growing number of studies are questioning the drug's usefulness.
We just decided between ourselves in November to write to Peter Duisberg and say, sorry to bother you. Are you for real? And if Lindsay were your daughter, what would you do? On November 11th, we got a big package, and he said, you must take your daughter off AZT immediately or she will die from it like Kimberly Begalis.
That is AIDS by prescription. You get immunodeficiency and you die from the tox. That is AIDS by prescription.
When AZT became widely available in 1985 and 1986, I cautioned my patients not to jump on the bandwagon and start being treated. I didn't want to see my community poisoned by an ineffective therapy.
I think in retrospect the dose that we started with AZT was a dangerous and poorly tolerated dose.
Nobody wants to realize what was the real effect of this overtreatment. That means that we killed a whole generation of AIDS patients.
In 1996, David Ho announced highly active antiretroviral therapy.
Also known as the cocktail, because the treatment combined the newly developed protease inhibitors with older HIV drugs, such as the chemotherapy drug AZT.
That was a revolution. What was an 100% fatal illness now could be treated.
The AIDS medication today is not that toxic than it was in the early days. And it's a potent drug regime. That means it kills almost everything.
I play around with treatment interruption because I think the drugs are toxic. And if I do the drugs continuously without interruption, I think that they'll have a cumulative damage.
In the years that we've been using the cocktail, we've found that there are lots of side effects.
In South Africa, I spoke to a couple of pharmacists specializing in HIV treatment. How often do you see side effects in patients? All the time?
Almost all the time.
We saw the lipodystrophy, the buffalo humps at the back of the neck, then the lipoatrophy, which is the loss of fat in the face and the arms, giving people a very gaunt look.
The risk of heart attack seems to be increased in people on these drugs.
With what we have now, the side effects eventually are going to outweigh the benefits.
So patients really do better for the short term, but in the long term, they die also.
In 1994, Audrey Serrano tested HIV positive. While initially healthy, she was prescribed AIDS drugs which nearly killed her and left her scarred for life. In December 2007, after multiple negative tests, she was awarded $2.5 million in damages.
Some people are very fortunate. They don't have these side effects, but many people do. So prolonged treatment is impossible.
I know people that are like horses, have no impact with some drugs, no side effects, and somebody else falls apart.
The new generation of antiretroviral drugs are less toxic. They can be more tolerated. But for how long? I agree. We cannot give a treatment for life. It's not like insulin. You know, it's something which is toxic.
Has a patient ever died from the side effects?
Yeah, sometimes it happens.
AIDS drugs are all classified as black box drugs. A black box drug is the most severe warning that our FDA will put on a product. It means you could die taking this because other people have died taking this.
My sister Joyce was my best friend. She's a great mom and just a very lively person.
In 2003, Joyce found out she was pregnant with a second child. She was offered an HIV test as standard prenatal care by her obstetrician.
She called me at work and she was like, I got something to tell you. And I was like, well, what is it? She said, I'm HIV positive. So I took a deep breath. I said, well, it's not the end of the world. And she said, well, Nell, I met this doctor today and he's a specialist. And he says that there's some medicines I can take that'll keep my baby from being HIV positive.
Nevirapine. Warning. Severe life-threatening skin reactions, including fatal cases.
One morning she was covered in these welts and this rash. It was all over her face. It was all over her chest, all over her arms, her hands.
When they're talking about a rash that can kill you, they're talking about a drug that targets the actively replicating cells in your dermis, in your mucosal layers, in your intestine, and stops them from working. And what happens? Goodbye skin.
I would never take them. I don't have a problem with other people taking them. But I, as Griselda Kananda personally, looking at the side effects that they come with, looking at the toxins that they present in my body, not now, not ever.
I have patients tested in 1985. They were all advised to take treatment, but they declined the treatment for different reasons. Because they didn't want to take toxic drugs because they were feeling well at that time.
And how are they doing today?
They're still living.
Healthy?
Yeah.
Once we came to the conclusion that this was the drug that was causing this problem, irregardless of what the HIV was going to do, she was going to come off the drug no matter what.
If taking her off the drugs meant that she could sleep through the night and be happy for 6 months, that would be worth it rather than live in agony for 2 years or 12 months.
You hear a lot of doctors, you hear a lot of educators in AIDS, you hear a lot of people talk about it is probably the drugs that are going to kill us before the disease does.
What are the drugs doing to the bodies? They're putting the bodies into coffins.
Before my sister started taking the drugs, she was healthy. After she started the drugs, she developed an allergic reaction, which made her look like a patient with full-blown AIDS. She was admitted to the hospital. She continued to spiral down. And within 37 days from her first date of taking the medicine, she was gone.
When news of Joyce's death reached the NIH, emails were exchanged between the director of the AIDS division and an ethics and safety officer. Ed, there was fulminant liver failure resulting in the death on this trial last week. Ouch. Not much we can do about dumb docs.
They're cynical enough to introduce drugs that they know will have toxic effects and will carry a certain mortality, and they know that the life of the drug before this mortality becomes too obvious to ignore is, say, 2 or 3 years, and they work out what the sales are going to be in those 2 or 3 years, and then they know they're going to have to reduce the dose.
Joyce has 2 sons. Jamal he's in his senior year of high school this year. Sterling will be 4 years old. And in my mind, they were robbed of their mother.
If someone's going to be giving me a diagnosis of certain death in 5 to 7 years, I want scientific proof. This isn't a religion.
My interest in questioning and breaking and exploring does HIV cause AIDS is an instinct to liberate people from a death sentence that isn't theirs to carry.
After we took Lindsay off AZT, her weight did go up.
Within a couple of days, the leg cramps went away.
And her physical body seemed to be doing pretty well, but it was like she was disturbed. She was agitated very easily.
Her lifespan changed all the time after she got to be 2 years old, and then she'd only live to be 5.
When she got to be a little older, then they said, oh, she might live to be 7.
Yeah. But definitely wouldn't live into double digits, and that's just was our life then.
Although Lindsay was on AZT for 22 months, she made a full recovery from the pernicious side effects. Lindsay will be 19 in October of 2009.
Because it's been surrounded from day one with so much emotion, so much fear, so much psychology, so much drama, very few people are capable of looking at AIDS logically.
We can be exposed to HIV many times without being chronically infected. Our immune system could get rid of the virus within a few weeks if you have a good immune system.
If you have a good immune system, then your body can naturally get rid of HIV.
Yes.
If you take a poor African who's been infected and you build up their immune system, is it possible for them to also naturally get rid of it?
I would think so.
It's very easy to get people to think the right thing if you get the right on the tablet the first time. But once something's on the tablet and you got to erase it and put something else, it's very hard to get people to think differently.
The victims of HIV and the dedicated professionals combating it deserve our sympathy, compassion, and respect. However, at journey's end, I find myself perplexed, bewildered at times with an overall feeling of dismay and sadness. I found a research community in disarray over the most fundamental understanding of HIV, all the while presenting a monolithic public posture of authority and certainty. Bluntly stated, we have tests that prove nothing, remedies that kill, and statistics manipulated to the point of absurdity. 90% of global HIV corresponds to areas of great poverty and squalor. Ironically, while we may have been pursuing a phantom killer, a shape-shifting assassin, perhaps the real enemy has been hiding in plain view, clear as day, and as old as time.
We can be exposed to HIV many times without being chronically infected. Our immune system will get rid of the virus within a few weeks if you have a good immune system.
If you have a good immune system, then your body can naturally get rid of HIV.
Yes.
How can we say that HIV is the cause of AIDS when we don't know, based on current tests, whether or not anybody diagnosed positive actually has HIV?
Some people are very fortunate. They don't have these side effects, but many people do. So prolonged treatment is impossible.
Called CD4 helper T cells.
What the fuck's he mean? What's it in there?
It uses its membrane.
What does he mean that they're not factor? Where is he coming from? There's factors for everything.
To the membrane of the cell. And then it
there, that cell virus grows and exports the cells. Gallo isn't going to change that. It's probably in the circulation.
Finds other CD4
He's
cells.
Remember
there is an ongoing battle. Cells die. The body will replace those cells and will die. But there's not an infinite capacity to do that.
Cofactors are important.
This
really
battle between HIV.
How people get ill, why they get ill.
What is asked of an AIDS journalist is to deny an existing reality, which is a strong, growing body of dissent on a scientific question.